Go ahead, pilot. Awesome. Yeah, let's make tracks, and if there's anything exciting, we'll reach out, but that slope is our goal. Thanks.
Thank you.
So if you are joining with us from shore, we are on the NOAA ship Okeanos Explorer with the Deep Discover ROV. We are diving in Nitinat Canyon off of Washington. My name is Alexis Weinig. I am a biologist with the U.S. Geological Survey and acting as the biology lead for this expedition.
And my name is Paige Koenig, and I am a geophysicist at Western Washington University, and I will be your geology science lead for this expedition and this dive. And like Alexis mentioned, we're in Nitinat Canyon, and we're kind of right inside of the canyon, and we're hoping to get to a steep slope of a north-facing wall. And we're hoping that that steep slope will allow us to see some hard rock geology and maybe a few outcrops. I know a lot of creatures like to hang out on those surfaces, so we're hoping to see large biodiversity in this area.
And we are underway with the EX2301 expedition. So this is the first expedition for the 2023 field season aboard the Okeanos. This has been a shakedown as well as an express exploration campaign, and so we've spent a number of dives at the beginning of this expedition getting the ROV ready and equipped for the rest of the season. So the ROV pilots have been accomplishing engineering objectives and testing out all of their systems. There's a new camera on D2 this year that they have been continuously testing that can take really high quality still images, and we have started to do some more science objectives. We were able to collect 4 biological samples yesterday, which was really great, and we're hoping for similar accomplishments today.
Definitely. Yeah, really looking forward. It looks like we're already seeing some creatures in view. We passed through a large number of jellyfish on the descent, if you were with us. That was really beautiful to watch. We've seen a couple of fish. Looks like there's a crab.
Yeah, we saw these jellyfish yesterday also. And if you have the stream up on shore, then if you look over on the second stream, it's the Sirius view, so the ROV that sits above D2, and you can see the jellyfish kind of bouncing around above us. And they're of the genus Predation. We saw a lot of them yesterday also, but we were much deeper. Not much deeper, but we were at 1,700 meters yesterday, and today we're at about 1,100 meters. So it's interesting to have those same jellyfish in this canyon as well as the canyon we were in yesterday, but at a different depth range.
Yeah, definitely. And today our dive path is going to be a little bit different than the kind of orientation that we've been doing. For the past couple dives, we've just been kind of going just kind of vertically upslope from bottom to top. But today we're going to kind of start on the bottom and then kind of move laterally while also kind of moving upward across the canyon wall. And hopefully that'll give us a neat perspective on the biota there.
Yeah, we were able to remap this portion of the canyon with the shipboard multi-beam last night. Our mapping team is up all night while we get to sleep, and they map the seafloor to give us new, fresh bathymetry data, which is seafloor mapping data to plan our dive for the next day. And so we were able to use that bathymetry to locate where we see really high slope, which is indicative of hopefully some hard bottom and areas where we have benthic communities growing. We have a nice crab here.
Hello, crab.
we have those lasers on, which are 10 centimeters. So the carapace of the crab looks to be about 10 centimeters in length. So we have these really amazing cameras on D2. So sometimes things look really large, but the lasers help us keep things in perspective, if you will. That's great.
Oh, wow. That's really cool.
I think this is a type of spider crab. We've seen these a couple times on these various dives so far. They definitely are okay in both the sedimented areas, and then also we see them kind of up around on the slopes as well.
Yeah, and we saw one a couple dives back that was missing some of its legs. It actually only had 3 legs on it, but this one looks like they have all of their legs on. That zoom in is really beautiful. And the current in this canyon looks to be pretty strong. You can even see some of the motion through the water column. And also on the surface there are kind of these like ripple marks indicative of the water that's kind of flowing through, which is also great because it brings in a lot of nutrients.
Yeah, and I think when we first got to the bottom before we were live on audio, Paige mentioned you could kind of see, and you can see it now, things kind of being pushed by the current that are lighter weight, so that helps us to, that allows us to be able to see that there is a current, because we're not down feeling this, and the ROV team sometimes can detect it with their vehicle being pushed, but it's not easy to determine what the pace of a current is on the seafloor, but we definitely can see some movement.
Looks like we're approaching some more fish.
And another crab.
Oh. Hello, fish.
Ashley Maranzino, who we have on board with us from NOAA Ocean Exploration, says that the fish on the left is a long-spined thorny head.
I remember seeing some of those when we were offshore Oregon, and we haven't seen one in a while, so it's nice to see them again.
See a couple. There's another one.
Hello.
And we can see that we're starting to move up a gradual slope. What we looked for in our bathymetry is kind of the steepest slope we could find that was operationally doable. And so as we continue to go up slope today, we should see an increase in steepness. So we'll be interested to see kind of how that plays out. We had some really great outcrops and features yesterday that we also guided that dive using bathymetry data and specifically slope data, and so hopefully our instincts are with us.
Yeah, since this is a canyon with a large kind of current that allows kind of the lighter particles like silt and clay to be transported really easily, and so that can create some like heavy sedimentation that can kind of cover up those harder outcrops. So hopefully, like Alexis mentioned, that the slope and that uplift will kind of eradicate a lot of that muddier sediment. Yeah, and Chris Ma in our science chat, one of our onshore scientists, just noted that we're not seeing too many brittle stars so far. And in our past couple dives, when we've kind of approached bottom, we've seen a lot of brittle stars. So this is an interesting difference to some of the dives that we were doing south along the margin.
Yeah, that's a good point. Same with almost all of, we normally have been seeing a number of echinoderms. So we've been seeing sea cucumbers or holotherians and brittle stars and even some true sea stars on the sediment. But here we're really not, at least unless we zoomed in, we might, but we're not seeing larger versions of those. We have a little...
Oh yeah, some rocks. Got a little rocky region. Curious what this is.
I might take a quick zoom on it if you guys are...
Yeah, that'd be great. Interesting. This sediment almost looks to be kind of like disturbed. I'm curious if that's... Yeah.
Not quite hard bottom, but
interesting.
Definitely some sponges and
some...
That's good. Thanks, guys.
Yeah, thank you.
So a little bit of exposure starting here, but not quite.
Yeah, not quite that rocky outcrop we've been hoping for.
But it really shows that even a little bit of something that's even slightly consolidated, you start to get growth, animals that are settling, like these small sponges, and then there seems to be some anemones. We've got a couple shrimp also.
Oh, yeah.
And there's a brittle star arm.
Oh, yeah. Kind of peeking out. Yeah. Yeah, I'm not sure if it's the same type of sponge that we collected yesterday, but I remember when it came off the ROV, I was the one that got to pick it up. And I was really curious as to what it would feel like, and it was indeed quite spongy.
Yeah,
Can report.
I don't think these are the same as the one we we collected yesterday, but I do think they would also have that spongy quality. They look like demo sponges. We have a buconid gastropod down on the bottom right, so a type of snail that's cruising along the seafloor, leaving a trail. You can also see the like tubes of sediment are likely excrement from organisms that eat the sediment and then absorb the nutrients out of it. So often a decent number of echinoderms, so holothorians, sea cucumbers will do that. So they are kind of like Paige made the analogy to worms earlier in the cruise, where worms on land will filter nutrients out of sediment. It's a similar process.
Looks like those sea stars are really appreciating all the little crevices kind of in this mound.
Yeah, the brittle stars.
Yeah, the brittle stars. Oh.
Looks like we have an anemone with its tentacles retracted.
Interesting.
Yeah, and I think just to the right of here, there's a chitin, which is a type of organism that will latch on. Has chitinous plates. Oh yeah, just there you go, just on the right. Has chitinous plates on its back. Oh, just to the right, Pilot, there's that little coral sticking out of the sediment.
Oh, I just missed it. Oh, I see.
Okay, no worries. So this is a umbellula, so it's a type of sea pin. So an octocoral that has these long tentacles, these long polyps and tentacles. They'll anchor themselves in the sediment and then are able to stay upright like that.
Another thorny head, kind of leading us along.
Here's a nice little rock.
Ooh, a rock kind of in a interesting, like, depression.
Yeah.
Oh, that's really neat.
Oh, yeah, lots of these thorny heads.
Oh, yeah.
Looks like...
Just kind of hanging out.
You think some of these rocks might have fallen from above?
Yeah, that definitely looks like rock fall, especially given the kind of depressions that are surrounding them. They likely fell and have been continuing to erode away that sediment surrounding it.
Oh, this is interesting.
Yeah, and
what's on the top of this rock? What's that
orange?
Oh,
that orange thing?
I'm curious.
I can't...
Oh, I think it's debris. Yeah.
Oh.
I thought it might have been a big octopus. Tricked
So
again.
deceiving. And Jenny Waddell, who's one of our scientists on shore with the Olympic Coast National Marine Sanctuary, has just brought up that there's a lot of glacial moraine in this region as well. So about 12,000 years ago, when glaciers were a part of this landscape and kind of retreating northward, they left behind kind of these piles of rock that they were transporting. And so some of this could be some of that glacial moraine that was left behind 12,000 years ago.
That's cool.
Yeah. I love when geology just connects to the history.
So glacial moraine means it's rocks that were trapped in the glacial ice?
Exactly.
Okay.
Mm-hmm. And then as it kind of melted, it just leaves it behind.
We have some anemones growing on these outcrops, which we saw a lot of anemones yesterday's dive in Quinault Canyon, just to the south of here. A lot of flytrap anemones, as well as a couple other varieties. They definitely seem to be happy in these really nutrient-rich canyon environments.
Mm. Are those shrimp in the water column? Those little orange?
Yeah, they're probably little mysid shrimp.
Okay.
They're really quick little guys, so I'm not sure if we'd be able to.
Yeah, and lots of marine snow like we were seeing in Quinault Canyon yesterday.
So, Paige, can you give us a little overview of how submarine canyons form? So we have canyons on land, right, like the Grand Canyon.
Mm-hmm.
Are they different than the ones we have under the ocean, or are they formed in the same way?
Yeah, that's a great question. So, yeah, like Alexis was talking about, we have the Grand Canyon on land where large volumes of water kind of erode the bedrock, creating these steep cliffs that form the canyons that we see today. And those submarine canyons are formed in a similar fashion where we need some sort of erosive factor, including like large volumes of water or mass transport of sediment, usually in the form of turbidity currents that kind of displace the sediment around it and form these continuous canyons. And Nitinat Canyon is kind of right outside of where the Salish Sea kind of comes out of the Washington coast and kind of makes its way toward the Pacific Ocean. And so it's likely that some of those currents have created the pathway through Nitinat Canyon that transport all of that sediment and large volumes of water into the abyssal plain.
Awesome. Thanks for that
overview. Yeah, if you kind of look at, like, even on Google Earth, you can see all of the kind of canyons that cut through the continental shelf along the coast of Washington and Oregon, making up some really beautiful bathymetry, which is cool.
And there's also really amazing canyons on the east coast of the United States also. And one of the things that we have been able to continue to document with the Okeanos, but also with other research expeditions, is just the amount of life that calls submarine canyons home. So the terrain that is created by those geological processes really makes a space for these benthic communities to really thrive. And so we often find really dense deep sea coral communities in submarine canyons, as well as sponge communities, which create a habitat for other organisms to live. So these are animals that are sometimes referred to as ecosystem engineers. So they create an ecosystem by living there themselves. And so it's really great when we can have documentation of that, and we have a lot of national marine sanctuaries that have canyons in them because they're such productive ecosystems. And so we have the Olympic Coast National Marine Sanctuary just to the west of us during this expedition, but there's a number of national marine sanctuaries that are working to protect the life that lives in these habitats.
Yeah, and also that flow of sediment can also add some more nutrients to the system, which also allows for that diverse biology that we can see in these canyons as well. And we can have Jenny Waddell, who's one of the scientists who works with the Olympic Coast National Marine Sanctuary, talk more about this, but Nitinat Canyon is also of high importance to the Makah tribe, who live close to these waters and use these waters for fishing. And we're going to have a couple live interactions with them later today and hopefully learn more about why these waters are important to them. So yeah, lots of people interested in our work here. Ooh,
I think
looks like a long...
Maybe an eel pout?
Looks really long.
Bigger eel pout?
Yeah. Ooh, shrimp.
Yeah. You can see this shrimp is modified more for swimming as opposed to kind of crawling along the bottom. It has those swimmer legs that are so gracefully undulating to keep it afloat. So certain shrimp are adapted to kind of crawl along the bottom or live more on the benthos, and then you have other shrimp that spend most of their life cycle in the water column, so up off the seafloor. There's so many jellyfish in the Sirius camera.
Oh my goodness! Oh wow!
If you have a chance to switch and look at feed 2, you'll see that there is just an abundance of these Paralia jellyfish above us.
They're just loving this nutrient-rich water.
Yeah.
Very cool.
And a little bit ago, Paige mentioned the term live interaction. So what that means is that that's something that the Okeanos really tries to incorporate into our science expeditions, is interacting with the public when we can. So we will do essentially like a ship-to-shore interaction with a classroom or with a community group, so that we can talk to them from the ship live about what we're doing and answer any questions that they might have about what it's like to be at sea or what types of animals or geology we're seeing. And it gives us a chance to really connect with the communities that are interested in the waters that we're exploring. Here comes some outcrops.
Oh, yes. Some more sneak peeks.
Sneak peeks, I like that.
Looks like a sponge kind of hanging out.
Yeah.
Oh, these rocks look different than the ones that we've been seeing. Can't wait to get a closer look.
This looks like a glass sponge that we saw at least within the same genus yesterday, Ferrera, which is a really delicate sponge that kind of has these lacy or kind of textile looking features.
Yeah, and as expected, this rock outcrop looks to be pretty heavily eroded and a little bit crumbly as well.
Oh, and so what behind us is actually a dead, possibly partially dead Faria, the same kind of sponge, but possibly has fallen down or collapsed. So we'll take a look at that afterwards. So you can see this one has this really bright white color. Mm-hmm. So that is an alive sponge that's continuously filtering the water through it to absorb nutrients.
Oh, there looks like there's like clams or something surrounding it.
So
do you know
I think those are brachiopods.
Brachiopods.
Which aren't quite clams, but they have, they do have two shells, but they are a whole different phylum.
Okay.
So whole
Thank
different
you.
type of, but they look a lot like clams.
Cool.
And we can see there's some brittle stars that are intertwined within this sponge. You can see the arms coming out, probably some type of ophiocanthid, so they have those spines projecting off of the arms.
And do you think they're feeding on the sponge?
Probably not actually the sponge itself, so I doubt that they're eating the sponge, but they are probably using the sponge as a way to get up into the water and able to kind of possibly steal some of the nutrients that the sponge is also trying to filter. So it's all about, getting up into that water versus being kind of submerged in the sediment where they might be smothered a bit.
Right.
Yeah, so we can see just above it, this is a probably a really large Ferrera that has either fallen off from above or has just become covered in sediment, so it's not able to filter as effectively, and they can't sustain.
It's huge.
It is huge, which gives me hope for what's to come.
Yes. Another sneak peek.
So we will, if we can just take kind of a, we don't need to stop, but take a quick zoom. Thanks, pilot. So you can see how it's more brittle and kind of like breaking off, and it's really covered in this sediment, so it's still creating habitat for other animals. You can see there's a lot living amongst this, but you can see that that sponge has started to degrade, and we can't really know how long this has been here, but it could be a very long time.
Yeah, it looks to be really covered in this sediment debris.
But there's lots of brachiopods. There's a gastropod, so a snail on there as well. A number of shrimp look like they're calling this home. Little shrimp hotel. That's great. Thank you, Pilot. And there's a big anemone living up there.
Hello, jellies.
We'll keep heading up to see what we can find as we get upslope.
I keep getting more and more excited. The more upslope we go, I'm like, what's going to be there next?
Look at them.
Oh, my goodness.
Jellyfish take over. I mean, it's a lot.
Yeah.
Oh, this eelpout's kind of wriggling around in the sediment.
Interesting. That's cute. Oh. Interesting. It just looked like it gulped something down.
Oh, might have. There's something kind of dark on the rock up in front of us.
Oh, yeah, interesting.
Wonder if it might be an urchin.
Ooh. Oh, wow.
Or maybe it's some type of anemone. Then that looks like a pretty solid rock, huh?
Yeah, it does. Okay. Yeah, especially looking at it closer. It does definitely seem to be just some rock fall from above or some more of that glacial moraine. Oh, interesting. It does
look like an anemone.
Yeah.
I think it might be one of these tumbleweed anemones, but a different one. So we've seen them on the sediment before, but this could be a liponema, but attached to a rock. So I think they have the ability to both attach, but also kind of just hang out on the sediment if they don't find a place to attach.
Oh, that's amazing. Looks like he's joined by some shrimps on the right and left. They're all over the place.
So these are cnidarians, or in the same family as, or in the same group phylum as the jellyfish we're seeing, or any corals we will see, but these have a mouth in the center of these tentacles. So all of the projections are called tentacles, and they have stinging cells that they use to catch prey and then transport to their mouth. And it's a really beautiful one. It's a different color than the ones we've been seeing on the sediment also, which were kind of a lighter pink. Some brachiopods also on these rocks. So brachiopods are kind of crazy. They were thought to be extinct for a long time.
Oh, really?
Yeah, and then people started realizing that they're still alive in the deep sea. And there's also a corallimorph on the left.
Hey, yeah.
Our good friend.
Yeah, we've been seeing a couple of those.
We collected one of those corallomorpharians a couple dives ago when we were a little more south, but it looks like the same type of corallomorpharian.
It's just amazing that even just something as simple as a rock fall can provide a whole habitat for a variety of creatures.
Oh yeah,
that's really amazing. Even that fish is having a good time.
Yeah. There actually seems to be a decent amount of these thorny heads here.
Yeah.
I wonder if they use this kind of rock habitat as a place to find food, since there's all these little shrimp that live around. Maybe use them for some hiding spots if they can get in any of the crevices.
Right. Ooh, exciting!
So the ROVs have sonar on the front of them, so it's a way of looking through the water kind of before you can see there. It'll show if there's hard targets in front of us, and so when you hear the pilot say that they see something 30 meters in front of us in the sonar, it means that there is likely something hard, whether it's a rock or even a wall. Sometimes it's a fish. Letting us know that there's something promising in the near future.
It's like another crab on screen.
It's like another one of these spider crabs. We've also seen, I'm not sure they're the same species, but at least a couple different size classes of these fish. So we have some that are a little bit bigger than others.
Yeah. And over on the left, there looks to be some sort of rock rubble that's kind of cascading down this slope, indicating that it's eroded away from a harder rock body. And a lot of the rocks that we see kind of on our dives and along this coastline are usually pretty unconsolidated sediment, meaning that it's pretty loose rock. And if you're there for a couple dives back, we tried to pick a couple up, but they unfortunately crumbled under the ROV's manipulator arms, and that's just because it hasn't been as heavily compacted, so it just makes it harder to kind of extract a sample.
And Kelly Markello from the California Academy of Sciences says she thinks that this looks like it might be a lithodid crab, which is a type of king crab. We do have a holothorian down there, maybe our first one.
Yeah, wow. That's surprising.
And we do have a nice amount of bottom time today, so we're only diving at about 1,000 meters, which means that we'll be able to stay on the seafloor for the majority of the operating day, just because we don't need to spend the time coming up from, say, 4,000 meters, which can take a long time to get down to and then also back up to. So operations permitting, we should be able to have a nice amount of time on the bottom.
Yeah, and I forgot. Do you remember how fast the ROV can descend through
So they
the water
normally
column?
go up and down about 30 meters a minute.
Okay, nice.
Yeah, we have a holothurian, and then also a little hermit crab.
Oh, good eye, yeah. Oh, small fish.
Yeah, there's even, see that little one up under the
Oh,
rock?
wow, yeah.
And Bob Carney from Louisiana State University has said that this is a latmagonid holothurian, or sea cucumber. So these are those animals that will eat the sediment and then filter out the nutrients, and then they release. So when you see those tubes of nutrients, that is the sea cucumber excrement.
It's so cool that you can even see it move through its central body. It's so neat.
These ones that are transparent really give you a look into the anatomy without having to dissect them.
Yeah.
So this would be the front end or the head of the holothurian. Those are feeding appendages kind of on the rock there, and then you see all the little parapodia, or the legs that it's using to kind of maneuver around. They don't have any bones, so these are invertebrates. So it's a water vascular system that allows them to move. So they're pumping water through their body to move all those little appendages.
Is that why they look so squishy?
Yeah. That's why they are squishy.
They are squishy, true. Oh, yeah. Interesting small fish.
Yeah.
You're going to have to ask Ashley, our fish expert, what this fish could be. Oh, Meredith Everett in the science chat saying that this is a young thorny head.
Oh, okay. So those orange and white ones we've been seeing are thorny heads also. So this is a juvenile.
Aw. Cute.
She said they're gray when they settle out, and then with really dark fins, and then they change color as they grow.
Oh, that's neat. Yeah, maybe if we could do a poke test on one of them. I think that they are probably likely heavily eroded and might just crumble, but if we could try maybe some of the sturdier looking ones, it looks like there's some kind of up top that look a little bit more compacted, but that would be great.
So, Paige, what is a poke test?
Yeah, great question. So since, like I mentioned, a lot of these rocks are pretty loose and unconsolidated, and so what I just requested the pilot to do is just kind of tap one of the rocks with the foot of the ROV, and this will kind of, or I guess the manipulator arm in this case, just to kind of see if it is a hard rock that we're able to pick up, or a loose rock that wouldn't be easy to sample. And seeing, oh, yeah. Yeah, it's all just kind of unconsolidated. Yeah, thank you for giving that a shot, though. Yeah. Yeah, and this test is nice for the
I can see
ROV
that
team, so they don't have to.
swimming.
Oh yeah, so that they don't have to set up full positioning for sampling, but we can just kind of test it out beforehand. Thanks, pilot. You can kind of see that some of these exposed boulders have undergone a lot of weathering, so when the minerals inside of the sediment kind of react with the water surrounding it, the iron will kind of oxidize and turn that dark color that you can see in some of the rocks. And oh, is that a cliff?
Or a larger rock?
A large rock. Oh, look at this outcrop.
Lots of holes.
Oh,
in
this
it.
is neat. Yeah. So kind of like we saw in this poke test, these rocks are really susceptible to weathering. So any kind of poke or disturbance will just kind of indent the rock. And you can kind of see that in this outcrop as well, with all of the separate holes that are likely an erosive feature.
Do you think that could have been caused by some type of fluid flow? They're very round.
Yeah, I would be curious to kind of get a closer look. Yeah, I think it could be very well remnants of fluid flow.
There's also a coral on our left.
Oh, coral. Yeah, this is really curious looking. It's almost kind of like, I don't know the right word to describe it. But yeah, just some sort of heavily eroded slope. Can't quite determine what it is. Carbonate!
Oh, really? Is this a...
Yeah, this could... Yeah, so our expedition coordinator, Thomas, is also a fellow geologist, and he mentioned that this kind of erosion could be carbonate-esque. I'm not too familiar with that formation, but I trust his opinion. Yeah, this is really interesting.
We're going to have the RV team take a closer look so that we can get some nice footage, maybe be able to help us determine if it's more of a carbonate or a mudstone or another geology term.
Huh. Weird.
There's definitely lots of biology living amongst it. They
Yeah.
like these little holes to hide out in. We have some squat lobsters in there. Some different types of shrimp. Definitely some sponges. It's a nice big white sponge on the right. Definitely some hydroids growing as well. Looks like a nice type of euplectellid sponge, so a glass sponge where it has that mesh on the top. So sometimes they grow that over and then
Yeah, we have scientists in the chat. I've just been peering into kind of all of those series of holes, and we have Bob Carney and Meredith Everett saying it looks like bioerosion, meaning kind of bivalves will...
On the left, there's that little pink coral.
Oh,
when
yeah.
you guys have a chance, that would be a nice zoom.
And those holes could be created by bivalves kind of inserting themselves into the soft rock face, creating those holes. Oh, wow. Oh, this slope is awesome. This is so cool. And it's amazing because, like, you know, canyons on land are, you can just drive to them, but submarine canyons, you have to take a whole ROV down there. So it's cool to be able to peer into these canyon walls that can show us more about the geologic past. And I think we also got a request to look at that sea star, but it looks like we're kind of heading to those corals, so...
Yeah, I think we'll start with looking at this little pink coral, and then we'll look at the sea star and the other fan when we have the chance. So this is a type of soft coral from the family Nephtheidae, sorry, blanked. And Meredith Everett has identified it for us as Jersemia julipacaridae. Julipacaridae, which I believe is named after a person. So these corals are a type of octocoral that does not have a hard axis or skeleton growing underneath it, so they just have small sclerites or calcareous elements that are embedded within the tissue to give them some hard structure, and then they are just essentially filled with water. So if we were to, we're not going to request this for a collection, but if we were to collect it when they come up to the surface, that water is all pressed out of the body, and it's just kind of a gelatinous blob. But you can see those 8 tentacles around the polyps really beautifully in that image. So Pilot, our other Zoom requests are the white sea star and the orange coral, so up to you, whichever you'd like to go to first. Thank you.
And given the amount of weathering that we can see with some of the darker kind of rock outcrops on the top and to the left, and also the amount of sedimentation that has kind of settled onto the surface, and the large number of kind of biologic holes, we can assume that this structure has kind of been here for a while.
Yeah, so I figured that, so the pink octocoral we just looked at must have been named for Julie Packard, who is one of the people that was instrumental in the Monterey Bay Aquarium So the Packard family is a large funder of that aquarium and the research institute that partners with it. So I have a feeling that due to her work in conservation and ocean advocacy that she had that coral named after her.
Oh, wow.
Yeah.
That's impressive. Love women in in science. It's amazing.
So here we have this nice white sea star. We'll see if we have Chris Ma in our chat to help us identify this sea star, which we've seen a couple of today. This is our first nice close look at it. This one has 5 arms. I think this is something we might have seen yesterday, but we'll wait to get some confirmation from him. You can see the little tube feet reaching out from underneath, which is how they move around. Also how they can bring food to their mouths, which is on the underside of this sea star. Their mouths are right in the center, but on the ventral side.
This guy looks to be kind of free of sediment cover. I wonder if he's been on the move for a while.
Or if there's a mechanism that they have for keeping themselves from being covered. Yeah, so Chris chimed into our chat and said he believes that this is Amphoraster, one of the early forcipulates, so related to Pisaster and Asteria, so an early lineage of sea star.
And Chris Ma mentioned in our science chat that this region is almost reminiscent of old Mesozoic seascapes, which the Mesozoic was millions of years ago, about 252 to 66 million years ago was kind of the Mesozoic geologic period. But it's almost reminiscent of that period with all the brachiopods. I like that analogy. Untested but interesting idea. I agree.
And then we'll take a look at this coral, which Meredith Everett from NOAA had said would have formerly been known as Swiftia, which is what I would have ID'd it as, and now is Calistephanus. This is a change in the naming of this, in the taxonomy of this coral, is due to some recent genetic work from Kathy McFadden at Harvey Mudd, and Andrea Quattrini at the Smithsonian Natural History Museum.
Oh, that's so fun. Look at the
Yeah.
shrimp.
We've got a shrimp, and there's also a capryllid amphipod. So in the left top, there's like a little projection coming off that's pink, but looks a little bit different than the coral itself. So those are amphipods that will attach their hind legs and kind of hang off of a coral or a rock like that to feed. And this is another type of octocoral that does have a skeleton that runs through it, so it has a more hard structure underneath where the tissue is attached with the polyps. We're getting a great close-up of those polyps open with the 8 pinnate tentacles around the mouth. And this shrimp is likely not eating this coral, but more just using it as a way to get off of the substrate. There's another shrimp. And Meredith said that these corals often have caprylids, so those little amphipods on them, and that the color of the capryllid often matches the color of the coral. Yeah, thanks, Pilot. Yes, let's do that.
Woo!
We have another Ferreira sponge coming up on our left. We've looked at one of these today, so I think we'll keep heading on our track. Oh yeah, there's a couple of them.
Looks like we have 2 more of the thorny head fish and maybe an anemone? Question mark? Also a considerable amount of rock debris next to it.
I think that's another corallimorph.
Oh, okay. See, I'm really trying with the biology identification. But I'm glad you're here. Corallimorph.
It is it.
Lars.
They're an interesting group that is related to like stony corals, like hard corals, but they don't make a calcareous skeleton. So they're kind of a little bit different.
Yeah, and as we continue up the slope, this geologic structure is really interesting, I would say, for a couple of reasons. First, the holes that are kind of in the rock wall are really interesting. We were thinking about whether these can be biologic.
Pilot, that coral, that white stick to the left would be an interesting target to zoom on. Thanks.
Whether these holes were perhaps biologic, created by like bivalves, kind of burrowing themselves into the side of the soft sediment. But like our expedition coordinator mentioned, it could also be some sort of carbonate, which could come in the form of limestone, which kind of forms when calcite kind of precipitates out of the water, and the water containing dissolved calcium, which could be a biologic or non-biologic process. That's something we can't really tell at the surface, but it's interesting to ponder as we traverse across this landscape.
Here's one of our big jellies that's been hanging out above us, but now coming into the view of the main HD camera. Nice shot of under the bell.
Yeah, wow. That's really cool.
Yeah, better door than a window, but just trying to steal the shot.
Mm-hmm.
And we are currently at around 1,045 meters depth in Nitinat Canyon off of Washington. We'll be working our way up a slope today to around 900 meters if we don't get distracted too much.
It's just so much fun.
Oh, this crab's missing one of his claws.
Oh, no! That must make it hard to pray. PR. See, this is me just trying to narrate the biologic side. I feel like I should maybe try to take a step back from trying.
No. I think you meant to prey upon.
To prey upon, not P-R-A-Y. P-R-E-Y.
So when we are set up, we're going to take a look at this white coral, which is a different type of paragorgia. So we saw the very characteristic pink bubblegum paragorgia yesterday, and this one has a white tissue, but the polyps might be a light pink color. Meredith Everett from NOAA has identified it as Paragorgia eutlenix, which is a species that she's seen a lot through these other express cruises or other express expeditions that have happened over the last couple years. So we can see those polyps all the way extended. Yesterday, and there's a nice chitin on the rock at the base also. Yesterday, when we saw the large Paragorgia colony, it had most of its tentacles retracted, so we couldn't really see how it might look when the tentacles and the polyps are protruding like this, but this is a great example where you can see how each polyp is oriented to the rest of the colony. And there's another little colony right above it. You can see a different base, so two different colonies of this same Paragorgia species. Wow, look at those angular.
Yeah, those like rock
know.
structures that are
Yeah.
kind of angular like that. Yeah, they are eroding in a very peculiar way. I'm noticing the angularity, but also the orientation almost looks like they've been uplifted, which is something that we would expect to see along the continental margin where a lot of thrust faulting kind of takes place that uplifts these sediments. And
Just don't say that. This looks like the same Paragorgia that we were able to just image, so we'll take a quick zoom on it, but then keep going. That white in the center might be of interest for a snap zoom. This looks like another type of sponge.
wow, that's beautiful.
Yeah, really interesting shape. Kind of has that matrix over the top. There might be sometimes you have small crustaceans that live inside these types of sponges, these glass sponges. There's definitely some crustaceans on the outside.
yeah, kind of peering in. Jenny Waddell said that they were calling them quesadilla sponges for a while. That's funny. I like that a lot.
Abigail Powell says that they got an ID that this might be Afrocastille's vastus. Afrocalistes. Sorry, not Afrocastiles. Afrocalistes.
And it looks like we're seeing a lot of more of these like blockier rock bodies and kind of migrating away from those kind of hole-esque outcrops. Bob Carney in the science chat just said that because we were pondering whether those holes were some sort of erosion from limestone, but he's also saying that the carbonate could be derived from methane, and methane seeps, and the degree of methane in the water column has been found to be high along the Cascadia margin. So this would make a lot of sense for where we are, North Washington.
Yeah, the process of methane, of methagenesis, so processing of methane by microbes, can create what's called authigenic carbonate. So it's a byproduct of that microbial process that generates just a carbonate that is often kind of pocky like that, and it's something that we see in places with a lot of chemosynthetic activity. And even if a seep dies off, so if the seepage stops happening from the seafloor, that authigenic carbonate is left over and can create habitat for other animals.
Interesting. I wonder if that's what we just saw below.
And Pilot, I have a collection request, but it can be one when we find one that works for setup, is one of these white sponges. It's going to be best to use the suction sampler because they're pretty brittle, so it works to just kind of suction a piece off of them. It'll break up, but that's fine. There's also a coral to the left. Yes, please. To the left, kind of your left corner. It's right above the fish. We can take a quick zoom on it when you approach, and we'll decide if we need a closer zoom. Yeah, so this looks like another of the octocoral we saw earlier, so if we can just get a focused image, then that should be good. See some more of those shrimp that are hanging out on this coral. But we have a hagfish on the left, I think. Oh, that's a jellyfish. All right, we'll keep climbing up this slope as we continue for this dive. For the next little bit, I will be narrating solely without my co-partner Paige. She's going to do a live interaction with a school that is interested in this area that we are exploring, so that's a great opportunity. There'll be a little less narration just so that we're not over inundating the control room with audio as they do their live interaction, but we will continue to operate this dive as we head up this slope. So our pilots are setting up to sample this white glass sponge in front of us. This is of interest to scientists that study sponges and the marine sanctuary that's nearby. They have discovered a number of new sponge species around this area, so the best way to be able to actually identify down to species is to have a collection in hand, so to be able to look at the spicules and other structures within the sponge tissue. If we can get a nice zoom, you guys normally do this, but we just want to make sure we get a good image of this sponge before we sample it. Nav, this is watch 1. Which canister are we on? Which suction canister are we on? Awesome, thank you. Yeah, that looks great. Great job, pilot. Thank you. So we were just able to collect a sample of a piece of that glass sponge. Abby Powell was able to let us know that they found 2 different species of this sponge in the Olympicos National Marine Sanctuary, which is nearby. And so the way that they're able to ID these is to look at the spicules under a microscope, and there's 2 different types. The structure looks different for those 2 species. So that's a great collection. We were able to collect it into a suction sampler. So there's 6 canisters that are stored kind of on the middle of the ROV, where that tube leads to a canister, and we're able to capture whatever came in through that suction tube. That is a big crab. And then we had a question in the chat about where are the samples that we collect deposited. So where do they go when they get off the ship? The biological samples go to the Smithsonian National Museum of Natural History in Washington, D.C., and then the geological samples go to a repository at Oregon State University in Corvallis, Oregon. We've got a really big crab here. I think this is a type of spider crab. You can see we had our lasers on, which are 10 centimeters, and so this is a crab that has a carapace that is bigger than 10 centimeters. Looks to be missing one of the front pinchers, which we saw earlier also in a different type, or a different crab, maybe the same type. Oh, and we have a small one on the rock as well, next to it. That might be either a juvenile or a different species. And what's really great about the repositories is that when the samples are deposited into those, they're publicly available, so any scientist is able to request a piece or that whole sample to either do some type of analysis or look into what that species might be or what that rock sample might be able to tell us about the region. And so this is a community-based exploration effort, meaning that we request input from the community members who might be interested in this type of research, and then they're able to participate either on board, like Paige and I, or they're able to participate from shore and then access those samples after they're back and deposited. Yeah, great. You can see the spines on the back of the carapace of these crabs. They're really distinct, and this is a great specimen to be able to get a close-up on for people to be able to ID later from just the video. Yeah, we have a small little juvenile crab. Possibly from the same family, but I am not a crab expert, so I don't want to assume that, but they definitely have similarities in their carapace. Okay, so Kelly Markello from the California Academy of Sciences says the bigger one looks more to her like a lithodid or a king crab. And then the little smaller one that we were just looking at might be a spider or a snow crab. So thanks for that, Kelly. There's also some squat lobsters that are on this rock that later in the dive, as we continue to move up, we'll take a look at. But yeah, we've got a couple more of these sponges coming up, like the one we just were able to sample. We will keep making our way up to try and cover some more ground. We have our lasers on, which are 10 centimeters, so we can see that this sponge is at least 30 centimeters across. All right, pilot, when you're ready, let's keep going. Yes, please. Sorry about the audio stuff for you. Yeah. Yeah. Looks like we have a hagfish right in center. Kind of slithering away. It's okay if he gets away from us. We have more of this very holy rock. Earlier we were curious if this might be, oh, there's a like pink stalk coral just to the right of center. Can we take a zoom on that? Oh yeah, that was the one I was actually. We can take a quick snap zoom on the one we're focused on now. But the pink one in the lower left. Yeah, this looks like another one of these Paragorgia that we have seen now, I think, twice on this dive. We've seen a lot of those thorny heads today. You can see the polyp starting to retract on this coral as it kind of senses our movement. This is a Paragorgia yetlinix, which Meredith Everett had let us know earlier. They've seen a decent amount of these on different express cruises across the express campaign. And we did not see this one yesterday, but we saw a different species of Paragorgia when we were a bit deeper at around 1,500 meters.
Yeah. So it looks like it might be deep sea sole, if I'm remembering correctly.
Up on the left,
Yep.
there's that, looks like a flatfish. But we're going to take a quick look at this coral, because I love a coral.
It's pretty interesting watching that hagfish try and
I know.
get into those little crevices.
Yeah.
And then we've got another little thorny head on a ledge right there.
Mm-hmm. Are those different species, or are they? Do you think they might be?
They might be different species. So there are two species that occur in this area: long spine and short spine. And you can tell them apart based on the length of their spine, if you could guess. So it's hard to tell. One of the species tends to be smaller, and the other one can get a little bit bigger, but they could also be juvenile. So it's hard to tell until you actually get a zoom in on those dorsal spines.
And while we have seen a lot of interesting things on these dives up these canyons, we have to balance the objectives of wanting to zoom on everything and actually making our way up so that we can see a decent amount of the canyon across the depth range that we're trying to traverse.
Yeah, and these thorny heads are nothing new. They're all over the place. I think long spine thorny heads are actually commercially harvested. So if you might see them in grocery stores if you're along the west coast of the United States. So they're related to the rockfishes in this area. So they apparently are the bigger long spines are also eaten. But I'll let Alexis talk about this coral now.
So this just has one kind of forking branch, which is interesting. It looks to me like a different type of Paragorgia. So we've been seeing this Paragorgia Linux today. That's the white, has a white morphology, but this looks to be one that is a different species of Paragorgia. Also has the same shrimp kind of associates that the other colonies have. You can see all of those projections are the polyps of the coral, and octocorals have 8-fold symmetry, so they have 8 tentacles around each polyp. That if we sat here and wanted to count them, we could see that all of them are identical in that way.
So are there any distinguishing features for like Paragorgia that help you tell that what this species is?
One of them, so let me see when the polyps retract. That helps. So now I'm going to change my mind. So it doesn't look like, so Paragorgia generally retracts its polyps all the way into its tissue, and they're normally, it has a pretty, I'll call it a chunky tissue. This looks to be like this Swiftia-like coral that we saw earlier, just not in the same fan morphology, but the way that the polyps retract and they're just still on the surface helps to identify it more as a plexaurid, so in the family Plexauridae versus a Paragorgidae.
Okay.
And then also the color can be, we don't like to use color too often, but there is a consistency with color for this group where you have the axis of the coral being that like corally pink color, and then if you looked at the polyps themselves, they were a little bit more of like a yellow color. And so that can help with identification, at least getting between those two groups. Paragorgids do not have that like yellowish color tint to them. Yeah, and that was a really nice caprellid amphipod hanging off the side, and that was something that Meredith had said earlier when we saw one of these plexaurids, that those amphipods are often found on this type of coral.
Okay.
And they have the same like coloration, so whether that's what they're eating or they're able to somehow kind of adapt that same camouflage color to look like they're just a coral polyp. And Meredith Everett had let us know that this used to be considered Swiftia, which was is a genus of octocoral, but it has recently been, the taxonomy has been reworked, and it is this one's hard for me. Calistophanes. And that's thanks to work done by Kathy McFadden at Harvey Mudd and Andrea Quattrini at the Smithsonian. Oh, you want to take a look at this fish?
We can if there's time, but I'm also okay moving along.
Okay.
Yeah. I appreciate the effort, though. Thank you.
Oh, we have another. We're going to keep going. I can't zoom on every coral.
That one does look chunkier. I wonder if that's a Paragorgia. Paragorgia. Yeah.
Yeah, so this looks like I think Ashley said a sole.
Yeah,
type
I think
of
it's
flatfish.
Yeah, so it should be I think it's a deep sea sole.
Wow.
Based on that coloration. So this is another one of those. It's a type of flatfish similar to a flounder. So it's got both of its eyes on one side of the head. So that's not the way that they're born. They're born with both of their eyes kind of like normal fishes. They swim around straight up, and then as they mature and they settle down onto the bottom where they're going to live the rest of their life, then the eye actually migrates over the top of the head, and that's how you see them now. So it's a pretty cool transformation.
Yeah, it is. He doesn't seem like he's quite camouflaged for his habitat. But
No, he's not ready for the sediment area. He needs a little bit more mottled area, I think.
I'm going to hop off and leave you guys with Ashley for a little while. I'm going to go grab lunch, but I will be back. So keep tuning in with us, and we should hopefully be seeing lots of exciting things.
Now, so here we have a sea cucumber. You can kind of see some of those little divots in the sand next to it that look like piles. That's probably some of the sediment that it's eaten and then just moved through its gut. So that's its little poop trail probably. But yeah, that's a good zoom. And I think Alexis is getting lunch. And so if we need to keep making headway, then we can do that. On the left, it looked like maybe a type of acorn worm. Those orange things. It's pretty cool. Yeah, you can tell that there's a lot of stuff living here. All of the sediment looks like there have been many detritivores eating along the bottom and making those little sediment trails. Mm-hmm. Yeah, and fortunately, I am not a geologist, and I don't know which type are going to be the most interesting rocks or if they're from this area or not. So unless I have somebody chime in on the science chat, then I think that rock looks a little large for us. Unfortunately, both of our geologists are in the middle of a live interaction talking to some people shoreside, so they can't chime in with their opinions either. We do have somebody in the science chat that says, "Get all the rocks." I appreciate the enthusiasm. Unfortunately, we will leave many of the rocks back on the seafloor. Yeah, that's unfortunately what we were seeing earlier on some dives as well. That's a new term for me. I do like that a lot. Consolidated mudstone. We were able to get one earlier. It was attached to a corallimorph that we collected, so we did get Paige's rock sample that way, which was good. Yeah, this is another type of glass sponge. It's different than the one we collected, but I think that I don't have any requests for zooms or captures yet. So if we need to keep making headway, then by all means you can keep trekking along. Okay, perfect. Cool. And Chris Ma is saying that these are euplectilids, a type of glass sponge. Yeah, and Abigail Powell just chimed in, so hopefully once we get a zoom in, she'll be able to give us a little bit more information because she is a sponge expert. Yeah. Yep, so we're zooming in on this glass sponge. You can also see a lot of life around it. So there are brachiopods. There are a lot of these little shrimps living on the sponge. And this is an incredible zoom. You can see the structure and why these sponges get that name of a glass sponge. They've got these silica structures called spicules that help create all that structure of the sponge. And if you pick these up, it kind of is like a fiberglass consistency. If you get them stuck in your fingers, hurts a lot. Do not recommend. And we've got another one of those capryllid amphipods hanging out on the top here. That's the pink amphipod at the top that looks kind of like a praying mantis. So we've seen those on corals. And Chris and Abigail Powell are both saying that this is a type of sponge called within the family Rosilidae. Great. This is amazing zoom. Thank you, guys. Yeah. Yeah, that dancing caprilid in frame. Oh, a couple caprilids. That's cool. Yeah. Yeah, and Abigail is confirming that. Unfortunately for these sponges, it's very difficult to identify a lot of them from imagery alone. So usually you have to wait until we get them in the lab. And this, so we've got a squat lobster there, and then it looks like a transparent sea cucumber. So we'll see if Chris Ma is able to give us more information on this guy. And then if we have time, Abigail Powell says that that sponge would be of interest for sampling if we're able to. Yeah, I think just a little piece of it. We don't need the full sponge. Oh, yeah, and then Alan Collins is, oh, yep, I see two, yep, in current and ex-current. Yeah, I agree, Alan. Alan Collins is saying that this is probably a tunicate of some sort. Yeah, I think they do. They look to be the same species, or at least they should be. Yeah, I think the bottom one would be okay as well.
Please pardon the interruption. Your conference contains less than 3 participants at this time and will be disconnected in 2 minutes. If you would like to continue, press star 1 now, or the conference will be terminated.
I think that looks good. Yeah.
Please pardon the interruption. Your conference contains...
Yeah, if it's small enough, then I think the whole thing would, oh, perfect. Looks great. Nice little haircut. Oh, great. Looks like we'll have some nice little associates with this too. And could I just get confirmation, was that the starboard and thank you very much. Yep, I'm good with all the labeling on that. Thank you, guys. Okay, sorry. Awesome. That was a great sample. Thank you, guys. Yeah, another one of those deep sea soils over here. Looks like another one of those glass sponges. I think it might be the other way around. And we've got another one of those thorny heads. So this one you can see really well. It's one of those long spines. The jellyfish, yeah, I mean, I think there are a couple of things that will eat them. I'm not sure exactly the species down here what will eat them, but like in shallow waters, sea turtles eat jellyfish. And sometimes like some fish will. They have to be pretty specialized so that they don't get stung in the process. That's a good question. I'm not sure exactly how shallow they get. I've only ever seen them kind of deep for this type. And it is interesting because they are planktonic, meaning they spend the entirety of their life suspended in the water column, just kind of more or less at the whim of the ocean currents. They can propel themselves a little bit, but they'll have their entire life swimming around in the currents a little bit. So I'm sure that occasionally they get into some areas where they're drifted up into shallower waters. I just don't know how long they're able to stay there or if they're with any sort of frequency found at shallower depths. Yeah. That is really interesting. We've been seeing a ton of these jellies over the last 2 dives. So if you are looking on the Sirius cam, which is, I think, channel 2 at home, you can see all of these jellyfish. They're in the genus Paralia, and they are just surrounding us in the water column. We saw a ton of them during the descent and during the descent yesterday as well. We also saw a couple of other jellies yesterday called big red jellies. So it's very interesting to see the sheer volume of gelatinous organisms in this area. I think it speaks highly to the productivity in the area. There's probably a lot of marine snow that is able to fuel them. They're able to get a lot of nutrients from the water column probably. I think if you pan a little bit to the right, there was a small white, possibly a sponge that was kind of spiky looking that somebody in Christmas was interested to see. Let me see if we can find it again. So I think it might be down now, so it might be out of view. Yeah, so like kind of center frame right now, I think. Yeah, but again, yeah, it's pretty small. It might be a sponge. Yes, it's kind of in that like dark patch in the center right there. There's. Okay, yep, thank you. So we're going through a watch change right now, and it is lunchtime aboard the ship, so we've been going through a little bit of shift change in general. I am Ashley Maranzino. I work with NOAA Ocean Exploration, so I am relieving the two science leads we have while they are eating lunch, and then we're doing some live interactions, talking to some people shoreside. Yep, that looks like a sponge to me. It actually looks like the one we just sampled, which is kind of cool. So, cool. Thank you for that. And we do have some of our scientists ashore have been remarking on how much sponge diversity there is in this area. So we've been seeing some of that during this dive, which is a lot of them have been glass sponges. We've collected 2 of them so far. So hopefully that will help scientists ashore get some identifications. And here we have some of these Paralia jellyfish coming through frame. They're the ones that are surrounding us in Sirius view. And now it looks like they're finally floating into our D2 frame as well. Yeah, it kind of looks like it's one of these jellyfish that maybe is inside out. I don't think it's doing too hot. I think that yes, I'm thinking, yeah, I think that, I think this would be we were getting a question earlier about like, does anything eat these jellyfish? Well, they might eat that one now that it's kind of pieces. So we have a new camera on board this time. We have a nice still frame camera. So our pilots and our video team have been working to try and get some images in that as well. So you might see some times during the dive where we're setting up to get imagery in the still frame or the still camera. So it looks like we've got another one of those corals down below us. It's interesting, kind of grayish thing in the center of over to the right. Yeah, if we can, but if not, feel free to keep moving. And it was, I think, to the right a little bit. Yeah, it's kind of a purplish gray to the right a little bit. Oh yeah, right there, to the right of the frame. Yeah, it kind of looks like it. It might be. See, yeah, it looks like a crinoid. It's interesting. Crinoids usually have their arms kind of extended more, but yeah, it looks like a purple crinoid. I think that's the first. Yeah, I think that's the first crinoid we've seen on this dive. And I think the ones that we saw yesterday, I don't think we saw any of this kind of purplish color. So crinoids are a type of echinoderm, so they're kind of closely related to some of the sea stars that we're seeing. I think hopefully we have Chris Ma on, and if he has any other ID for us. Yeah, this is a gorgeous image. So crinoids are kind of an ancient lineage. They've been around on Earth for a while. They're also called feather stars, and you can see why. It's really interesting that the tips of these arms are a different color. They're that white color. Gives it a very nice ombre effect. Yeah, there are a couple of There's some mysid shrimp around associated with this. Looks like there might be another shrimp kind of behind it on the ground. So you can kind of get a sense of why these are called feather stars. They've got all of their arms really look like feathers off of the central body of this crinoid. This is gorgeous. Yeah. Yeah, you can see all of these. So these are arms that will get used for feeding. So they'll catch particulate or floating debris out of the water column. Yeah.
We also got a request to zoom in on the arm tips, if that's possible. Thank you.
And we have a couple of experts in the chat that are, we have some crinoid and echinoderm experts. So we've got Chris Ma and Kelly Markello in the chat. So pretty. Awesome. Thank you so much. That was a great zoom.
Yeah, and after closely examining the zoom in on the tips of the arm, Kelly, one of our scientists on the chat, says that the arms look perhaps damaged and maybe are missing some of the tissue.
Interesting. So maybe they were nipped off by something looking for a meal down here. Yeah, very cool. Thank you guys for that zoom.
Yeah, thank you.
Well, I'm going to go grab some lunch. It is Taco Tuesday here today, so I'm going to grab some lunch, but Paige is back, so I will leave you with her.
Yes, hello everybody. Once again, my name is Paige, and I'm the Geology Science Lead, coming from Western Washington University. And I will try my best to help narrate the biology, but a lot of my interest lies in the geology and the geophysics, and it almost looks like as we're heading up a slope, we're seeing some of this rock debris that's been kind of persistent. And this rock debris, like I mentioned, is likely due to the softness of the rock bodies here. Yeah, it looks like an interesting hill, a nice trail. I'm curious if it's either biologic or I don't know. There seems to be like some white sediment that's kind of dispersed throughout the trail. I'm curious if any of the scientists have seen this as a biologic phenomenon. That would be my first guess. Looks like we're joined by several more thorny fish, as well as some jellyfish. Yeah, this is a really great wide view. We're really getting an idea of just how steep this slope is. Looks like we're getting a really nice zoom in on some of these Paralia jellies that we've been seeing in the water column. This is a really amazing view into kind of the underside of the jelly, under the bell. Ooh. Looks like there's some sort of crab that's kind of burrowed into that little hole. Oh, joined by several different crabs on the side of this cliff face. And it looks like there's a starfish located, kind of center. Yeah, this sediment is really pervasive. There's a lot that is accumulated on this steep slope. Oh, squat lobsters. Thank you, Chris. Right now I'm out here solo, so I do appreciate our benthic biologists helping me out here. And so right now we are continuing our way kind of laterally along this canyon slope. We're at about a depth of 993 meters in Nitinat Canyon. And yeah, we've already seen some amazing creatures. We've already seen some jellies. And there seems to be a lot of these thorny head fish kind of around. And these are some pretty spectacular geologic structures that we're looking at. In my opinion, I think it's still this pretty unconsolidated sedimentary rock with a couple of burrow holes that we can see. We're coming up kind of by where that thorny fish is sitting, where perhaps some creatures have been making homes in. Yeah, I believe so. We've seen a couple at depth. Yeah, so this looks to be an anemone. Has beautiful, vibrant pink colors. I think we've seen several of these already, but it's cool to see one further upslope, kind of nudged into these geologic crevices. That's a beautiful shot. Ah, yeah, that is what it looks like. Yeah, I'd be curious if any scientists in the chat have an ID on what that orange thing is on the left. But yeah, it's really beautiful. Yeah. Ooh, there looks to be some activity over on our right on the side of this rock. It's really, really nice seeing some of these like recently eroded rock surfaces. You can see the kind of darker rock kind of in center view can show some of the intense weathering that's really been occurring. These rocks are heavily, heavily weathered and cemented over. But it's cool when a block falls away, so you can peer inside to see kind of what the rock looks like on the inside. Yeah, and this location along the Cascadia margin is actually really, really interesting. Geohazard-wise, that's one of the reasons kind of why we've come out here along the Cascadian coast. Right now we're along a subduction zone boundary, which means we have an oceanic plate and a continental plate kind of meeting up together, and that oceanic plate is diving beneath the continental plate. And we haven't seen an earthquake here since about the 1700s, and so it's been a long time since there has been an earthquake, making it hard for us to study the structure of the surface at depth, and making it harder to predict geohazards for the future. So it makes a lot of this research that we're conducting and a lot of the research that scientists are conducting along this margin extremely valuable. It looks like we're coming up. There seems to be a couple more sea stars scattered on the surface. Some more of those holes that we've been seeing. Really, really beautiful canyon wall. Oh, is that a sponge? Yeah, just below. It looks like there's a sponge that we've been interested in. This expedition. And really great perspective on the canyon wall here. You can see a lot of the blockiness of the geologic architecture. And so those at home who are unfamiliar with kind of our ROV systems, right now the D2 or Deep Discover ROV is has two cameras. There's an HD camera, which is the one that you all are looking at, and there's also a still camera that allows us to take, or the videography team to take really amazing stills of some of the creatures that we see at this depth. And that lattice structure on the, oh, looks like there's something inside of the sponge. Can't quite tell what it is yet, but it's kind of peering out. Hello. Yeah, it looks like he... This guy looks very protective of the sponge. He felt us coming.
Yeah, it looks like another couple of those Paragorgia. So, hello everybody. We are back on after lunch and live interactions. My name is Alexis Weinig. I am a biologist with the U.S. Geological Survey, and I am acting as the biology lead for this expedition.
And my name is Paige Koenig, and I am a geophysicist at Western Washington University, and I'll be your geology science lead for this expedition. And right now we are at about 970 meters depth, kind of traversing the side of this canyon wall in Nitinat Canyon, which is offshore northern Washington. And we've already seen some amazing creatures, including...
Ooh, there's a white coral up at the base. Oh, it might not be a coral. It might be a sponge, but still worth.
Still, yeah. You have such a great eye for that, Alexis. Every time I'm like, what are you looking at?
The ROV pilots get it. After doing this for a while,
Yeah.
they just begin to spot things from what feels like nowhere.
Mm-hmm.
Some of the work I did during my PhD was in the Gulf of Mexico, and we were looking for type of coral called Paramaricia. It's a yellow fan coral.
Okay.
And I can tell you, I can spot those things from a mile away now.
Oh, wow.
We have a couple interesting things here. Right in front of us, we'll take a look. It looks like possibly a sponge. Or a coral. Who knows? That's a coral.
Ah, coral. Oh, that's neat.
Yeah, so this looks like a type of... I'll get a closer look once we can see the polyps. That'll help with identification. Oh, so this looks like a type of black coral. So,
Oh, wow.
yeah. These are called black corals because they have a black skeleton, but the tissue can be a number of different colors. This one seems to be kind of an off-white. The one we collected yesterday had more of an orange color to it, but this looks like to be, is this a possible collection pilot? Okay. Yeah, then let's take a look, but this is definitely something we would be interested in.
It's really interesting. It looks really like bushy almost.
Yeah. Yeah, it looks like there's a couple of them here. So this looks like a black coral that we haven't seen on this expedition yet. I'm going to look a little bit through some of our guides and documentation, see if I can get a genus for us, but it's definitely different than the ones we saw yesterday, where we saw what looked to be something like a Bathypathes and then Trissopathes. And this looks to be a new black coral. And we're at about 900 meters, which is pretty standard for black corals, so in a good depth range to find these.
Oh, and Jenny reminded us that the non-technical term is floofy.
Okay, so, and Meredith Everett's chimed in that these are chrysopathes.
Oh, okay. Yeah, I was curious about that too, Chris. You mentioned there's like a white ball that we kind of passed over, and I'd be curious to see what that was, but I think we're going to zoom in on this black coral first.
Yeah, Meredith said these are, you can find these in pretty big groups or patches, so that might be why we're seeing quite a few of them as we've come across one.
It's interesting that they've really cemented themselves into this pretty like heavily eroded landscape, latched onto some of the harder rocks that lie beneath the sediment.
Yeah. Yes. Yes. Yes, it would.
Oh, looks like there was something on the coral, which we might...
Yeah, it looks like a squat lobster is
okay.
living in the coral.
Oh, yes, hello.
Yeah. Yeah, you have one in a suction jar.
Oh, okay.
Did you guys sample something else?
This is the third sample, so I think that there was one sample that happened. I wasn't a part of that sample collection. Yeah.
I don't know.
The one prior to this collection?
Okay.
Correct.
Thanks.
Yes.
Yes, that would be great. This will be like the orange one you sampled yesterday, a little bit sticky, but hopefully it'll cooperate. Yes, it does. Yeah. Oh, there goes the squat lobster. Thanks, pilot. So we were able to just sample a black coral, a chrysopathes. We collected the whole coral, which actually will be very good for a taxonomic review. So having the whole colony will allow taxonomists to look at the morphology of the branching pattern as well as the base of the coral, which can be really important for identifying maybe how old that type of coral can be. Yeah, don't worry, it will all be used. That's okay. These can be in pretty dense patches, so it's not like we're taking the only one, and we're definitely going to use it for scientific investigation. Well, it's also that this is, like Paige has been talking about, it's essentially mud, so I don't think it was attached very hard to that rock.
Oh, I wonder what that's.
Do you think it is? Yeah.
That like spiky looking thing?
Yeah. That looks like another black coral. If we can just take a zoom on it. Sorry, just left of center. Looks a little.
I see.
Yeah, pretty translucent. Yes. Yeah, this looks like another species of black coral. So kind of this planar branching pattern. So not the big bushy, floofy ones, but just has branches coming off on each side. Looks like we might have come across a nice depth range for black corals. Now seeing a couple of different ones.
No, and this is a great perspective of the entire kind of slope that we're looking at, and you can just really see how eroded away everything is from this kind of bird's eye view. Yeah. Right. Yeah, let's see if we can pass by it again. I know that we had Chris Ma was interested, but if we can't find it, it's okay.
Maybe it was this anemone that...
Oh, yeah, it's
I
just
think it's
an anemone.
An anemone, yeah.
Thanks, pilot. Ooh, interesting white sediment over on the left.
Yeah, so that's where their foot was, right?
Oh, that's
so
where we grabbed from.
Yeah.
That's why.
Yeah.
Oh, I see, I see.
Oh, nice sponge.
Ooh. Very tubular.
I think that's another one of those quesadilla sponges down there.
I love that for a name. That's pretty much exactly what they look like. Ooh, there looks to be some interesting creatures on this rock face. Some of them seem to be kind of retracting and...
Oh, yeah, they might be... Oh, there's also a nice sea pen right at the base there. So that little stick...
There seems to be another trail kind of along this muddy bottom. I love spotting those.
Looks like we have some more of these chrysopathes, black corals. Jenny Waddell pointed out a kind of a shaggy barrel sponge that we'll try and locate and get a good close-up on. There's another sea pen to our left. I don't think we have. If we can do a zoom, that would be great.
And can you remind me what a sea pen is again?
Yeah, so it's a type of octocoral
Okay.
that doesn't normally secure its base to a hard surface. So they have what's called a peduncle, which will kind of bury in the sediment, and it just kind of holds them there. So you often find them in more softer sediment areas.
Oh, interesting.
And they normally are not branching, so they're kind of just like this pen shape. Kind of looks like a feather pen.
Yeah.
From back in the day.
Yeah. Oh, wow, yeah.
Yeah.
You can really see the base.
I think that's just some gelatinous stuff on top, but underneath
Oh,
the
okay.
sediment, there would be the little peduncle, and then as we pan up the stalk, we'll start to see the polyps as they splay out on each side. I'm curious from either Jenny or Meredith back on shore if you guys have seen these sea pens in this area before. We're at about 950 meters right now. So you can see through the translucent part on the left, there's a white line. So that's essentially the stalk of the sea pen. So that's kind of hard. Mm-hmm. It's calcareous, and then the rest of it is more gelatinous. And so when you bring them to the surface, one of the characteristics, or what we try and do, is keep them from snapping. So they're pretty fragile. You want to keep them in one piece. It'll help for identification. All right, so Jenny and Meredith said these are very common in this area. So this is Balticina.
Mm.
That's great. Thanks, video. Thanks, pilot.
Ooh. Looks
some other fish.
Yeah.
Yeah, different fish on the left. Some more of these chrysopathes. Like Meredith said, it seems that when you find one, you might find many.
Yeah, I'm curious what that fish is. We should ask Ashley. Oh, and looks like this is a sablefish.
Okay.
Really beautiful spotted colors on its back. You can see it filtering the water in and out. Also very common. And is that a sea cucumber?
Mm-hmm. We have a little yellow sponge right in the center.
Oh, wow, yeah. Almost looks like neon.
Yeah, like a bright yellow probably. Oh, I don't know what that... That's good for now. Thank you.
Thanks.
Oh, yeah.
Oh, wow.
You can really see kind of like how sheared off.
Yeah,
I guess how
this
steep
like
this
rock
is.
face, yeah.
Another one of these kind of tubular barrel sponges. Okay. And Pilot, those, we've been informed that those yellow sponges like we saw are on our collection list. So if we see another one like that, that'll be a target. Okay,
Sounds good.
So the ROV team and the video team are working on the settings for a still camera that's new to the vehicle. So focusing on something that's white helps with the color balance a lot. So they're going to take a minute and work together to take some images of this sponge. Oh, great.
Oh, cool.
So the full stream, or on stream 3 for the streams that are going to shore, you can see the still camera that is being focused on right now. So there's 3 camera streams that we stream to shore during the dives. There's the main HD, which is camera 1. Camera 2 is Sirius cam.
Yeah, the Sirius view.
And then the third one rotates kind of between a quad view of a couple other cameras. And currently right now we have it on the still camera so that you guys can see what our video engineer is focused on.
And it looks like there's something hiding out inside of the sponge.
Probably.
Yeah. Nice spot.
while we're going over it, we'll see if there's a inhabitant.
Oh, yeah. He's got a little...
Yeah, that's good. Thank you. Great.
Ooh.
Yeah, that
Indeed.
sounds good. Have we looked at this white star yet today?
I think we did a zoom in on a white star. I don't know, I might turn to...
Oh, yes.
Yeah, let's do that. Good eye.
Yeah, we'll start.
Right, yeah, definitely.
Yeah, that looks great.
Wow, it's so different looking than
So they
the other
were-
sponges.
Oh, Chris Moss said we don't need to look at that sea star, so that's good. And this is this lunar sponge that Jenny Waddell and Abigail Powell have been talking about. So we are going to set up to do a collection of the sponge. What's nice is it's a relatively small, contained collection, so we'll probably aim for collecting the rock or the mud that the sponge is on and get that whole specimen. Did they say that? Did they say that? Oh yeah, there should be a sample in canister 1. Great. Great. We saw the sponge drop into the suction canister, so that was an awesome collection by our pilots.
Nice job. And the
I like watching the replay.
me too.
pilot, there is a sponge just to our lower left that we would like to get a zoom on if possible before we leave this spot. That's okay. There's a sponge just to our lower left, like a white, like bubbly looking sponge that we just want to take a zoom on before we leave this spot, if you guys are able. There you go.
Oh, nice.
So Abigail Powell was saying that this is possibly a new species of Hexactinella that Henry Weiswig is describing. So we'll get some good images of it, and we'll keep an eye out for it as we continue to head up. There's also a nice little octocoral above it. I think it's one of those... what used to be Swiftia, now I have to learn this new name. Hold on. Callistophanus.
Oh, wow. So many tubes.
Yeah.
Really interesting.
That was great. Thank you.
Yeah, thanks.
Some more of those white Paragorgia. Lots of squat lobsters on this outcrop. We might want to, at some point, pilot, it doesn't have to be right this second, when you see one of those orange little squat lobster crabs that we can take a zoom on, that would be great. They're all over the place, so make your pick.
Yeah, I was just noticing on like the upper kind of right, there seems to be a mysterious black rock. And I don't know where that came from. It looks like it's been kind of displaced from wherever it was from. But I'm curious. It's really stark against the landscape, and it looks like there's some white lines that are kind of covering it.
Those are probably tubes from a type of worm.
Interesting.
That make that casing. There's also a little coral on it.
Yeah, this is really peculiar. So you can see it almost looks like large white clasts, which we describe as kind of if there are these like smaller kind of large smaller rocks within a larger matrix. We call those clasts. And this looks like some sort of conglomerate, but it's really strange. And I'm wondering if it's maybe one of the glacial sort of remnants that have been deposited in this area. So often, 12,000 years ago, there used to be glaciers kind of covering the Pacific Northwest, and as they retreated northward, they kind of left behind a trail of rocks from a bunch of different places that they kind of crossed over, and so this could be kind of one of those displaced rocks. And it looks like Jenny said that this rock has some sponges that they're interested in kind of encrusting it.
Yeah, so when you said the casts, what I think those are actually sponges that
Yeah.
are growing on the surface, and then there's also some brachiopods and some those little tube worms. You can see the feathers coming out. So those are little worms creating those white tubes. And then we have that pink octocoral, which I think is that other Swiftia-like coral we've been seeing. It's definitely a very different rock type.
Yeah, really funky.
Would you be interested in collecting this?
Yeah, I mean, we could definitely do a poke test and see if we're able to, but I think that this would be an interesting opportunity to sample. And yeah, it looks like it's about perfectly 10 centimeters across.
What would be really nice about being able to collect this is if it's informative for geology, we also will be able to, we call it pick the biology off of it. So we'll use all of those other organisms as collections as well. So it really multiplies our collection.
That's correct, Pilot. No worries. Thanks for trying. No worries. Thanks, Pilot. So what you guys just saw at home is we saw this kind of protruding rock nodule that seemed a lot darker than the surrounding sediment, and it seemed like a good rock sample to take back with us, but unfortunately it seemed to be too much, it seemed to be incorporated too much into the rock face behind it, so we were unable to pluck it out. And I think that that sample was also a really good representation. Pilot, we just saw a coral off to our right, if you are able to go back along that way. Thank you. And that looks like another sablefish hanging out, although I'm not positive.
I think that's a Mercurid, so one of these rat-tailed.
Oh, man. We try our best. At least I try my best.
So I wanted to see this
Oh.
one. Oh, it's another Paragorgia. Sorry, it just has more branches
Yeah.
than the ones we've been seeing. The one that's just below the middle. The one that's just below in the middle. We can do a snap zoom. It doesn't have to be a land.
It's really kind of fun as we like traverse the slope. Those kind of orange dots on the slopes like close up automatically, and it's kind of funny as we continue on. They're just like, nope.
I think they are possibly tube anemones. We haven't really taken a close look at them, but that's pretty. Yeah, so this is another one of these Paragorgia
Oh wow.
with the mostly white tissue, but they have the pink polyps. You can see the polyps retracting at the top of the, at the tip of the branch, or the branch kind of most to the right. And then you can see the extended polyps. There's a little shrimp on that leftmost stalk, too. You see the eyes?
Oh. Yeah, I just noticed them. It's
Pretty
kind of
good.
hiding in plain sight.
Yeah, pretty well camouflaged. I think we see these shrimp in association with these Paragorgia pretty commonly. Some of the larger colonies in the Atlantic also.
Interesting.
That's great. Thank you, Pilot. Did our rock sample not go as planned?
Yeah, unfortunately our rock was too cemented into the surface, which
Oh.
I know, a different kind of problem than what we've been used to
Yeah.
having. Usually we can't even pick them up just because they're so crumbly, but this one was too strong.
Also kind of curious since, let's see, I don't know, I think we were thinking that everything is kind of just sitting on the surface of this, but
Yeah, that's kind of indicative that perhaps this surface is much more consolidated than it appears. And it's likely that a lot of the rock that we're able to interpret from is kind of hidden beneath this layer of sediment that's kind of piled on top of it.
no, I think that's good. Thank you.
Yeah, oftentimes when a geologist is out in the field, they'll be carrying around a rock hammer, and that's mainly to just kind of open up the surface of the rock, get past all of the erosion and weathering to kind of peer behind it. But that's extremely hard to do when we're at this depth. Can't bring a rock hammer with us.
Seeing some more of those nephtheid soft corals, which we saw earlier today.
Is that light one in
Yeah.
the center?
We can keep going, Pilot. Thanks.
Ooh, we got a lighter color sediment
Yeah, it looks
kind
like
of on our left.
you think maybe something broke off.
That's what it looks like to me, is that recently there was like a chunk of the slope that just broke, and we're just now getting to peer onto it. the squat lobsters really love like the nooks and crannies
They
here.
do, yeah.
There's so many of them. Oh. Huh.
If you have the chance to look at Feed 2 or Stream 2 online, you can see the Sirius view where we have all these jellyfish that are still in the water column looking. The Sirius is looking down on D2. You can also see just how vertical this relief is that we're working our way up.
Yeah.
Well, this is a really interesting.
Oh, whoa.
I think this is a sponge.
Yeah. Accompanied by another crab.
Yeah. Let's take a closer look and we'll be able to figure out some more information about this.
Yeah, strange. The texture on it is interesting.
Oh, yeah. This is a coral.
Oh, wow.
So this is a primnoid octocoral. Has incredibly dense polyps.
Yeah, wow.
We've got some shrimp living amongst this coral also.
Oh!
And a fish.
Hello? Nope. Kicking up some of the sediment around us.
Let me see. And Meredith Everett says at this step this will be Peristanella ramosa. Oh, is it?
Wow. Oh, wow. You can really see so much detail on that
Yeah.
shrimp. You can like peer inside of its body. It's really cool.
You can see so all the coral polyps are kind of projecting out this group of coral. The primnoids don't really retract their tentacles or their polyps into the tissue like some of the other octocorals do. They kind of fold them up, so they actually have like really some of the sclerites, the calcareous structures are on the outside of the polyps, and so when they curl up, they look like armored. Like if you look at them under a microscope, they kind of look like they have like layers of armor on them.
Oh, wow. What do you think's causing this one to be so dense? Is it just the type, or is there like high nutrient availability?
It's probably just the species, so
Okay.
it's probably the growth form of this particular type of coral, but it looks really healthy, so it is probably the nutrients in the water that are keeping it looking so good. You're not even really seeing any bare branches. And also, I think the amount of shrimp on there are indicative that there's a lot of flow happening. They might be hanging out there to catch particles that are kind of getting stuck in the coral, and they're able to feed off of those small organisms or the detritus in the water.
Oh, wow.
we're going to come around and look at the other side. From one of the other cameras, the ROV team was noting that there's a lot of shrimp on this side also. You can also see the way that the coral is growing. It kind of has that arch shape. That's likely due to the current, so whichever way it's facing, it's kind of growing in that way to be able to almost create like a little net around itself and catch those particles in the water.
Oh, wow.
Oh, it's beautiful.
Yeah.
And we have our video team working with the still camera again also. This is a really nice opportunity to work on refining that focusing and lighting process. Meredith Everett is noting that this species is often full of brittle stars, but this one surprisingly isn't. So
yeah, we haven't really been seeing a lot of brittle stars
in
this region.
It's also, it's relatively planar, but you can see that one branch in the back is kind of branching off.
Yeah.
Curious just about that growth formation. Might have to do with the current direction or...
Oh, wow. That's really cool.
Looks like the polyps face down on this species, so that's one of the things that can help distinguish certain species or genus from each other is the direction that the polyps close with, especially in relation to primnoids, since they're not really retracting back into the tissue. And I think the one we collected yesterday was a Caligorgia, and it had its polyps closing upwards when we brought it to the surface. It's a nice shrimp. It's also not polyps growing on the main axis of the stalk, which is also a diagnostic characteristic, so where the polyps are growing. Ooh, nice squat lobster.
Oh, wow. Hello.
Yeah, I think it's at least one of them, so that's a good target to get a zoom.
Yeah, Jenny Waddell, who's one of our onshore scientists, is noting that the dissolved oxygen is super low here, and so on D2 we have these sensors, and one of them is the dissolved oxygen, and that could be the reason why...
Say that again? Okay, great. That's good.
And the low dissolved oxygen could be the reason why we're not seeing as many brittle stars here, which is some great insight. Thanks, scientists. Wow.
Yeah, it's a great coral.
That's a beautiful shot.
Great flying, great video work.
Oh, yeah. So those lasers measure about 10 centimeters across. So we can really, see how large this specimen is. Looks like 20 centimeters
tall.
Laterally, yeah.
Maybe about 40 centimeters wide.
Dang.
It's nice. Gorgeous.
Ooh, looks to be another jellyfish.
Yeah.
They've been kind of following us around here.
Another sea pen, which we've been informed are really common in this area.
And some thorny head fish.
Looks like we have maybe another black coral up in the right corner. So we are currently diving in Nitinat Canyon off of Washington, and we have been working our way up this side of a canyon wall from around 1,100 meters at the start of the day, and we're currently at 925 meters. We have been surveying this wall, and I think, Paige, do you have some insight about the geology that we've been seeing today?
Yeah, so as we kind of traverse this wall, we've seen a lot of heavily eroded and unconsolidated sediment structures, which makes sense because right now we're located along the continental margin where there's a high volume of sediment that's just kind of accumulated over time due to the subduction zone that's kind of located in that area. But kind of interestingly, from the rock sample that we took today, we actually found that some of this rock is actually a lot firmer, and this could just be due to the pressure temperature conditions that it was formed under and lithified from. A lot of this is likely sediment that has just undergone high pressure and temperature and been able to solidify.
Upside down, Jelly.
Oh, hello. And it almost looks like we're kind of nearing the edge of a recently eroded rock surface, super sheared off. You can see it's just straight up and down. It looks like someone just took a slice out of the cove, which is really interesting.
It's a great perspective where we're off the wall a little bit more, so you kind of see the
different layers.
Yeah, and you can get an understanding of just how steep it is, and you can even see that it's mud, right? Like
yeah. Yeah, usually when we're kind of looking at sedimentary rock, one of the major things that we're looking at is kind of the size of the particles that make up the rock, and so a lot of this mud is, as you guessed it, made of yeah, they look a little bit detached there. That one looks like it's kind of fallen from a steep slope. But a lot of this kind of marine mud is often really silty, meaning that the particle size is really small.
Yeah, so now it looks like we have maybe some
bouldery
rocks?
Yeah, exactly. Bouldery rocks is a good way of putting it. Sometimes we call it cobbles.
Cobbles.
Yeah, cobbles. Usually, cobbles are kind of rocks that have eroded down slope, and sometimes they can, depending on how far they've rolled, they can even be kind of round, and that's just from all of the angular edges just kind of being eroded away by its tumble fall or by the surrounding environment. And it almost looks like we're approaching like the top of something. I can't quite tell.
Yeah, we're definitely coming over. Maybe there's some more slope ahead of us, but there might also be like kind of a local high, if you will.
Yeah, that's a good way of putting it.
Yeah, it looks very like pebbly, gravelly here.
Yeah,
versus
on the bottom, it's much more silty and fine. Yeah, another important kind of thing that we think about geologically is kind of the energy of systems. And so oftentimes if you see larger cobbles, that could be indicative of a high energy system that was able to supply some of those larger, heavier rocks. And since this is a giant canyon system, that could mean that there was some sort of transport through the canyon that transported some of these rock, or it could be just rock fall. Can't quite tell.
Hey Nav, this is Watch One. We have a question. Are we looking at a southwest facing scarp / slope? Essentially, like which way is the wall facing? Thanks. Yeah. Yeah, so it's like a southeast facing scarp. Yeah. Okay, I know. It's like okay. Thank you.
Thank you. Yeah, you can see much more of this cobbly rock.
Yeah, would any of, I mean, I know we tried one, but maybe at some point try another?
Yeah, definitely. Yeah, I think with rock samples, we do want to try to get something that's representative of the surrounding rock. And so sometimes a cobble is kind of hard just because it's not adjoined to any of the basement rock. But I think that we should definitely try, if we do see one that looks a little bit similar, we can definitely try.
And Bob Carney says the cobbles look more like they might be glacial material here. So like Paige was talking about before with how when glaciers were melting in the past?
They were, yeah, definitely. They were retreating, and as they were retreating, they were melting. Yep. Leaving some of that rock debris here.
We'll take a... Okay. Yeah, I will agree with Nav on that.
Ooh. This is a nice field of coral.
Yeah, this looks like the black coral that we collected earlier.
Oh, nice.
That has been noted to kind of be patchy when it is around its... when you find one, you might find multiple kind of situation. Chrysopathies. And Meredith had noted earlier that the little ones might just be younger versions of the same species. We have a nice thorn head.
Is that a thorny head? I can't. It looks like we've been seeing those orange thorny heads, but this one has like an interesting color pattern. We'll have to rope in our fish expert here, Ashley. Love his big eyes. Able to peer into the dark water column. All right.
Oh, and he's off. Yeah. Pilot, there is a starfish, just not the little one close to us, but the one in the background a little bit. Yeah, looks like maybe a little flatfish.
Oh,
A very small one.
oh, he's so cute. Are these the ones that have like the one eye on one side? Oh, actually, might be able to see it. Oh, okay. Oh, so these are the ones that have both eyes on one side. This one has a really cool pattern. It's so intricate. Meredith Everett saying that this is a deep sea sole. Hope that's the correct pronunciation.
Yeah,
like,
sole.
Okay, cool.
It's a type of flatfish,
Okay, nice.
or a pretty common term for flatfish. We saw another deep sea sole earlier that was much larger. I don't know if it's the same species, but the other one we had identified by Thomas Monroe via Alan Collins at the Smithsonian at the National Systematics Laboratory, and the other one was a Microstomus bathybius.
Oh, wow.
And here we come to this larger sea star that Chris Ma was asking for a zoom on, so when you guys have a chance, that would be great. Right next to a nice little Paragorgia and Chrysopathes, so we got a couple corals as well as a nice big sea star here. And he's feeding.
Oh.
So this sea star is eating this sea pen that it's on top
gosh. That's so cool.
of. So these are predators, and they are corallivores, is what we sometimes refer to animals that eat corals. We heard a beep.
Sounds like we have someone on our teleconference line.
Greetings.
Hello,
Who could this be?
I know. As you've acutely pointed out, as all coral people seem to know by now, there are sea stars which feed on their animals. It doesn't seem to matter what habitat we're in, but in the deeps, there are certain families of goniasterids, which is a very large family of sea stars that feed on octocorals and coral and just cnidarians in general. A lot of them belong to the subfamily Hippasterinae, and this is the name-bearing genus Hippastereus. They are predators on a wide range of cnidarians, including sea pens, octocorals, which are part of the same group, but they also feed on sea anemones and many related animals. And the moment that we began seeing a lot of these corals, I knew we were eventually going to see something like this. It's almost inevitable. But basically what happens is they extend their stomach out, and they will begin to digest a lot of the prey item externally. Oh, wow, that's one of those. Oh, so this is actually a new, I would say that this is a new capture, because this looks like, I mean, it could well be another species, but it's probably Hippasteria californica, which it turns out is not endemic to California at all, so there's a lesson to be learned about what you name as a species. But this is that sea pen that Margaret, I think, commented on as being fairly common, and yet I don't think we've ever gotten an occurrence of this species feeding on this sea pen in this way. In shallower waters, Hippasteria phrygiana feeds on the sort of shallow sea pen Tylosarchus, the sort of fluffy orange one. And so, and in other places, we've certainly seen Hippasteria feeding on any number of, deep water octocorals. We've seen them perched precariously and literally chewing their way from the bottom to the top.
Oh, wow.
And, in some cases, some of these sea stars can literally be feeding on some of these coral prey for months, if not conceivably longer. So, this is a very gradual and slow process. But, the time involved for them is very different. It occurs on a much different scale than us. Just so that we're sort of, what you're seeing, what I'm seeing, a lot of the stuff that we zoomed in on. So we're seeing all of these spines, and I'm not quite sure why, but every corallivore that feeds on octocorals has big prominent spines. I'm not quite sure if that's to defend against other predators or if there's some way they facilitate predation. The other thing that we tend to see a lot of are little claw-shaped or clam-shaped structures called pedicelaria, and they kind of open and close when they're feeding. Again, I'm not entirely sure why. We saw these on the Hawaiian species several years ago. So, there's a lot of unexplained mysteries. But so it's feeding on this sea pen, and here we see another sea pen, which is probably going to become dinner, in a few months. Yeah. So, there are, there's a lot going on here, and I've described different species of other families of sea stars that feed, for example, on those Antipotherians that we've been watching. So I'm looking forward to seeing some of my other friends, if you will, some of the other species that I'm familiar with that have have literally climbed onto some of these, corals, and sometimes they don't necessarily even start at the bottom. They just climb to the top and work their way down, I think. But we haven't seen the whole process for things that are not hippasturines. Hippasteria tends to feed from the bottom and eat its way up. And in fact, a few years ago, we saw one in the Hawaiian Islands that fed, and it ate one of those primnoids that you guys saw just a few moments ago. And all of the capsules that the primnoids have were still intact. And we discovered that, in fact, it was actually inserting its stomach into each primnoid capsule and eating the inside.
Oh my goodness!
I mean, that makes a lot of sense.
And leaving the outside capsule, yeah.
It makes a lot of sense because primnoids have those really hard casings on the outside, so the stars have to have a way, essentially, of getting to the tissue, whereas these other octocorals are really just soft polyps all the way around. Wow, that's fascinating.
Oh yeah, so there's all sorts of stuff going on here, and the back and forth between predator and prey is a fascinating kind of exploration, especially if you look at long-term macroevolutionary trends. So, that's another story. Anyway, I see we've moved past the hippasteria, and we're moving up the wall of the canyon again. So I'll leave you to it, and have a good day, and thanks for at least one wondrous sighting of a sea star today.
Thanks, Chris.
Thank you.
Thanks very much. Bye-bye.
Bye.
It's always great to have our onshore scientists kind of clue us into their specialties.
Yeah, it helps a lot with just trying to get to the details of some of this exploration. We can bring just a couple scientists on board with the Okeanos Explorer, and so it helps a lot when we have scientists on shore who can participate watching from their offices or even their homes, and they can be we have a science chat where they can write in comments or suggestions, and then we also have that teleconference line so they can call in and talk to us on the ship.
Mm-hmm. It looks like we're looking at another nice sheer cliff that's been, again, heavily eroded and has a bunch of holes and nooks and crannies for some of the creatures here.
It's so funny to me that you call them creatures.
Yeah, I know. Well, I'm always thinking, like, what could I say as a nice broad term? I guess I could say, what would be a better, more scientific way of saying? Benthic biology.
Or animals.
Animals.
It's cute. It's
Looks
very
like
geology. Very geo-lead.
Yes.
It's like me talking about, like, is this a rock?
This is a rock.
Yeah, you can say organisms, you can say animals.
Awesome. Another thorny head. Seen a lot of these around here.
Yeah, and I have a feeling that if we were able to really peer into all of these little crevices, we would see a whole bunch of life living within them. It's hard to do that, but I bet there's squat lobsters, there's small shrimp, there might be brittle stars tucked back in there.
Yeah, you can even see some of the squat lobsters now.
Yeah.
Kind of poking out.
Here's a couple more sponges. You have a white one just down in the center, and then looks like one of those yellow kind of lunar sponges that we collected earlier today.
Ooh, another crab I see.
Mm-hmm. So many holes in this rock or mud. I mean, this seems like mud to me.
Yeah,
Okay.
yeah, the way that it's eroding kind of looks muddier to me as well. Although there is kind of another one of those kind of darker rock nodules over on the right that we kind of like the one we were trying to sample. It's like little wedged in there.
Yeah, Jenna Hill in our science chats was commenting that this light gray rock face is interesting because it looks kind of freshly eroded. We were saying that earlier, that looks like some of these have kind of sheared off more recently than others is kind of how we could say that, just because of the coloration of the sediment.
Yeah, definitely. A lot of when you see kind of like the darker outside of the rock, that's usually because due to the weathering in the water column and the reactions of the minerals inside of the rock. And so when it shears away, it kind of leaves the true color of the rock, which is that lighter kind of mud.
And I think earlier today, when we were seeing one of these larger crabs, Kelly Markello from the California Academy of Sciences had suggested that this was a lithodid crab, so from the king crab group or family. We've seen at least half a dozen of these today.
Yeah, definitely. There's been a lot of them. It's amazing that they can just traverse over these steep slopes. It's like amazing to me.
Like, I mean, they have the fortune of anti-gravity underwater.
True, yeah, they are underwater.
But still, it's like completely vertical.
Mm-hmm.
It always makes me think about how cool like marine animals are.
Yeah.
Because you don't have crabs crawling up mountains or canyons on
definitely
land.
not.
And in our science chat, we had Chris Ma, who is an echinoderm expert, and Meredith Everett, who studies deep-sea corals, commenting to each other how neither of them had seen that observation of Hippasteria on a sea pen like that before. So it's a novel observation to see that specific kind of predation event. So we're getting some good still image shots of this thorny head fish with the still camera that's mounted just above the HD camera on the front of Deep Discoverer, the ROV. We're also in the process of doing a pilot shift change, so we're going to switch out our ROV team, and then we will continue with the rest of today's dive.
Yeah, I'll be curious. I'll be curious how much longer we have on the bottom, but I'll wait to ask before we have our pilots all set up.
Wow, look at that close up.
Oh my gosh!
looking into
Wow.
its soul.
Is that the reflection of D2 in its eye?
Yes, it
Oh,
is.
that's so cool. Oh, wow. All right.
Oh, now he's like, that's enough.
That's a little too close. Whew.
We have a sponge right to the left, too. Another one of those vase sponges.
Accompanied by a jelly.
Man, yeah, those Paralia jellyfish have been... We're seeing a little less of them now as we've come up from when we were...
Yeah, in the Sirius cam.
Yeah. I mean, they're still here, but they were just really dense.
Not as many.
When we were around 1,100 to 1,000 meters, there was a lot of them.
Yeah.
And we are currently aboard the NOAA ship Okeanos Explorer for the EX2301 expedition, which is a shakedown, and it is an express exploration campaign. So this is our eighth dive of this expedition, and we have one more planned dive for tomorrow.
Our last dive tomorrow.
Yeah. Already.
I know. It's gone by so fast.
And we will be making some decisions about where that dive will be later today, but it will be somewhere within Nitinat Canyon, and that will be, will put us in position to be able to make it into port to Seattle the next day, and then there is a public port event on Saturday, so not August, we're in April.
April 29th.
April 29th. There will be a port event in Seattle where the public is welcome to come to the ship at Pier 46. To tour the ship and see D2, the ROV, as well as Sirius, the other ROV, and kind of get to take a look into what it is to be on board and work on the Okeanos Explorer.
We're finally peering into this eroded wall that Jenny pointed out earlier, and you can almost see at the bottom the sediment that's accumulated from that rock fall, which is really interesting, and some vertical kind of breakage along those rock plains, just showing how unstable it is.
I feel like it's quite different from yesterday's dive, right? The Geology?
Yeah, definitely. It's definitely a lot different. Or I guess also similar in some ways where we're seeing unconsolidated mud. But also we're seeing more of that glacial erratic here, which is kind of new.
Yes, we've seen a lot of these thorny head fish. Mm-hmm. So we're kind of, I think your previous pilots communicated to you, but we're kind of just dog-legging or saddling across this face, just so that we stay on the slope. Yes, there was a lot more earlier today. You were in here earlier today, too.
familiar with Nitinat Canyon, told us that this is kind of interesting to look at this slope from a slope failure perspective, as in like seeing where the places of the rock kind of slumped away, is because there are more frequent about magnitude 6 earthquakes, so kind of middle range magnitude earthquakes that happen along this portion of the margin, the Cascadia margin that's kind of closer to...
Could we request a snap zoom on that sea star? Thanks.
Northern Washington, as opposed to where we were yesterday, which was Quinault Canyon, that experiences less earthquakes. Thanks, Jenna.
So we have a different sea star, a goniasterid, sometimes referred to as a cookie star or a ravioli star.
Oh, that's fun.
Could also possibly be feeding, but I'm not gonna say for sure.
Oh, those are really cool.
And a little squat lobster tucked up in that crevice also. Wow.
That's a really nice shot. You can really see the ravioli edges.
Yeah. The crimping.
Crimping.
I mean, I don't know if that's not a science term. I'm just, that's how I would describe that.
Yeah, it is what it looks like.
So Chris Ma has identified this as medaster, which is a type of goniasterid sea star. So thanks, Pilot. That's great. And that edge, Chris says that edge is
Crenulate?
crenulate.
Fancy. Looks to be just about 10 centimeters across.
Mm-hmm.
Exactly.
I mean, it is funny. I always think of them as larger.
Yeah. Yeah, when we zoom in like this, I imagine gigantic. I even think about that when we take samples, you know, and like when you're snipping them off like a coral or something and putting it in the box, you're just expecting this gigantic thing. But then when it comes to surface, you're like, oh, correct. We just have great video equipment.
Yeah.
And when we were looking at that kind of cleaved off rock face and of that sheared rock, you can't see it now, but hopefully we'll come across another region. Bob Carney just mentioned that you couldn't really see any of those holes that we can see on the surface here, which is indicative that these are definitely biologic, or most likely biologic.
Yeah, and Jenny Waddell from the sanctuaries also made a comment when we were talking about the shearing off of those cliffs and the likelihood of that and the effects of earthquakes versus down in Nitinat Canyon has some effect on the type of biology that can live here too. So you're probably not going to have really long-lived or corals or sponges that have been able to stay in place for a long time. So you're going to have some of these species that are able to utilize this more muddy habitat, but maybe not the ones that really need to latch on to a harder surface. Or they do that and then they just don't make it because they fall down with the avalanche.
Yeah, with the rock fall.
There's another one of those yellow lunar sponges in the bottom left corner.
Oh, yeah.
And a nice Paragorgia coming up.
On one of those.
Oh, can we actually
glacier
take a look,
rocks.
not at the coral, but to the left? There's like the white projections coming out. Thanks. I think this might be a type of sea cucumber
Oh, really?
that extends, yeah.
Oh, cool.
Yeah,
Oh, wow.
it's definitely a holotherian, so a sea cucumber.
Oh, super cool. I don't think we've seen one of these at all.
Or we have definitely not zoomed in on one, so I don't know how to say the name. I know how to spell it. It is Psolus, so maybe Solus? Solus? Or Solidae would be the family that they belong to, but yeah, very different type of morphology from the sea cucumbers we are used to seeing that kind of crawl along the seafloor.
Are they always branched out like this?
Yeah, so
Okay.
so they're essentially like settled. They are attached,
Oh,
kind of like
okay.
an anemone would be,
Mhm.
and then they extend these appendages out, and that's how they're able to feed. So you can see in the center the mouth.
Oh, wow.
And
yeah.
so if you do a time lapse of these,
Uh-huh.
so some of them they're shallower species. You can see them put their whole little arms into their mouth and pull them out. So
Oh
they
my
continue to
gosh.
feed themselves with those appendages.
Oh, wow. That's really cool.
And the tentacles will retract if they're like disturbed. So it's a very similar morphology to an idarian, but it's an echinoderm, so more closely related to sea stars or brittle stars. And right next to it would have been an anemone that closed up, but
Oh,
we would have
yeah.
had that very similar kind of morphology. Oh, or it's I think it's a closed anemone. Oh no, the one on the right is also the same kind. It's just closed up.
Oh, neat. That's a cool comparison.
Oh yeah, because you can see the little bits of its,
Yeah, they're like still kind of poking out.
and they're like hairy like that. So with anemones, you don't have those projections on the tentacles.
Oh, wow. That's really cool.
Yeah, that's cool. That's fun to see.
Yeah.
And right on that little rock. Yeah, and we have a squat lobster also that we're able to get a nice zoom on. There's two. There's one under the rock, and then we have those encrusting sponges and bryozoans on this rock, so it's just a wealth of biology.
Yeah, this is a great spot.
Yeah, you can see those tentacles kind of peeking out.
Oh,
Oh.
looks like he's popping back out.
Ooh.
Oh, that's so cool.
We'll see if it starts to extend.
Yeah.
Maybe it's not going to move fast enough
he's-
for our timeline. But
He's thinking about it, though.
and Bob Carney said that these types of holothorians are most averse in shallow water. So there's more species of them in shallower waters than down here in the deep. But I don't know if I've seen one personally before, so this is cool.
Oh, sweet.
That's great. Thanks, Pilot.
Yeah, thank you.
Lasers.
Oh,
So our 10 centimeter lasers, their
Oh,
bases
these
might
are
be
super
about
small. like-
maybe like 5 to
Yeah.
8 centimeters. It's also, I always am in awe of how inhabited, like if you find one really solid rock.
Yeah.
It's always, that is very expensive real estate.
Oh,
on the
yeah.
deep on the seafloor.
Yeah, they all seem to know and find those places. It's cool.
Here's another sponge. Oh, another one of those Hypnaster?
Oh, the sea star? Or
Yeah, the
coral?
sea star. Oh,
Okay.
the coral's Paragorgia, but the sea star, I will get it by the end of today. Hippasteria. that's what it's called.
Hippasteria.
Which was the one that we saw feeding on the sea pen earlier.
Okay.
Yeah, we've got another rock full of, just lots of encrusting organisms.
Oh, yeah.
Now, this is watch one. Could you let us know how much bottom time we have left? Okay, thank you.
Sounds good.
Looks like we have a...
Can't. Huh.
Maybe... Maybe a type of sponge? But it's
Yeah, I
like
don't
rolling
know.
around. Looks to me like there's like intakes like you would have for a sponge, but I'm not quite sure. Maybe somebody in our
Yeah.
chat has a better idea. Yeah, there's a hiding brittle star behind it.
I haven't been seeing very many, but looks like we just found one. Yeah, really interesting. Really gelatinous, clear. Oh. Oh, Liz Clark saying it's a tunicate?
Oh, definitely be a tunicate. Or it could be crashed something from the water column that has kind of made its way down to the seafloor.
Really interesting.
There had been a comment in the chat about from Jenna Hill about possibly aged. Oh, there's that coral, sponge.
Oh, yeah, the one that we were zooming into earlier.
Yeah.
That was unknown?
Yes, we are. And if it's something that we could possibly collect that would be of interest, it's probably best to slurp it, or to... Uh,
3.
so we've been told that this one's it'll be hard, but it's also brittle. So the suggestion is to slurp, but if you guys want to try grabbing it, that's also an option. I just want you to know what you're dealing with.
Yes.
I think that little section on the right might be a nice section to go for. Oh yeah, there's chunks in there. Oh, if you can get that, yeah. Take it. Yeah, nice. Great job. Everybody can take a deep breath. That was a great collection by our ROV pilots for another sponge. So I think that's our fourth sponge collection for today. And we have one coral collection. We tried to collect a rock, but it said no.
We did. It did say, no thanks, I don't want to leave.
And Abigail Powell's best guess for the ID on that sponge is Hexactinella, but it is one that will definitely need to be reviewed onshore by sponge experts like herself.
Great, so now it looks like we'll get to continue to traverse along this canyon wall. Right now we are at about 909 meters of depth within Nitinat Canyon, offshore northern Washington. Looks like we have about an hour or so left on bottom before we'll have to begin ascent. I'm excited to see what else we'll be able to see here. Ooh, is that another sponge?
Yeah, definitely looks like it. Or is that the one we just...
Is it the one that we just sampled? Oh, yeah.
Is it?
Are we underneath it? Okay, yeah.
We're like, nah, orientation.
Well, I was also seeing that fish was over there, but I don't know if it's a new one, but it must have been.
Oh, look
Nice.
at that, like, is that a fault?
A
A crack?
Yeah, it's likely a crack, yeah, especially because we're looking at this kind of unstable slope, so it's likely a vertical crack, but yes, I do get excited about this still. Any evidence of faulting is exciting.
Yes. We
Oh,
do.
mhm.
Yes, if you don't mind. Cool. Sounds good.
And we also got a request to take a peek into the direction that we're moving, if that's possible.
We have another one of those sea pens. Oh, what's this on the... Oh, is that a rock?
Oh, that darker rock,
Yeah.
yeah. Looks like another one of those large cobbles.
Yeah, I feel like whenever we kind of see one of these harder rocks, there's at least one of these Paragorgia on it. So I have a feeling if this was not so tectonically active, why do you get earthquakes?
Yeah, exactly.
Okay.
Yeah, tectonically active would be a good way to put it.
Then there might be more coral colonies growing on if there was harder rock substrate. Seems like every time we find one, there's at least one coral on it. So they want to be here. Oh, we have another
Ooh.
deep sea sole. So this flatfish up here. That's okay. We have seen that today. Nice big sponge
So big.
and a bigger Paragorgia
Ooh, interesting
very-
orientation.
flopped over. Pilots, are you able to get a sense of, is there much of a current down here? Okay.
Pilot, we also got a request to face the direction that we're moving in, if that's possible, but I'm not quite sure.
Or just take a peek at that direction, if possible.
Yes.
That's great. Thank you.
Ooh, looks like we have another kind of trail or a place where a rock just kind of tumbled down up in center, which is kind of fun. Another sponge.
There's our slope. Yep.
Nice.
Looks like we'll come around this feature.
This bend.
Have a nice big sponge. I think we've seen a couple of these today, but I don't know if we have a definitive ID. We'll see if Abby Powell on shore has an ID for us.
And last time we saw something at the center of the vase, so I'm curious if we'll be able to see something hiding inside. Oh, that's a cool perspective.
Yeah, it can be
That's
hard
great.
to peer down into these with our large ROV.
Yeah. Oh, that's cool.
Okay.
Oh,
Thank
that's
you.
great. Thank you. It's interesting to kind of see how this canyon wall has changed. It seems like we kind of started at bottom, and we kind of then when we went up to a steeper slope, we saw a lot of steep, lightly sedimented areas, and then we kind of approached this region with some glacial cobbles, and now we're kind of in a zone that looks to be a lot more heavily eroded than the place where we began, which is just kind of interesting as we traverse from northeast to southwest.
Yeah, this looks like a different kind of sponge where it has these really large pinnules coming off.
Ooh, shaggy. Oh, wow.
Yeah,
That's
really
cool.
has all of its defenses out.
Yeah, I wouldn't want to mess with this guy.
Looks like we have some little ophroids living in there.
Mhm.
Also some small shrimp on the surface. Some brachiopods.
It's accumulated a lot of sediment. Oh, Jenny's saying it should be clean inside, though.
Yeah, it's like the sponge we sampled yesterday. Remember, the outside has that, gets all the muck stuck on it.
Mm-hmm.
But inside they are pristine. Oh, a big squat lobster down there.
Oh, whoa. Yeah,
Coming out. Yeah, Jenny Waddell says that's a good example of benthic habitat, or pyogenic, sorry, habitat. So habitat being created by something that is living or was once living.
Yeah, very true.
We have a couple little corals like the ones we've been seeing throughout this dive, this white paragorgia, and then a pink octocoral. So Abby Powell said that these larger vase sponges can be tough to ID without having the sample in hand. The big vases are all lysocene sponges, and they're different from the sponges like the ones we collected with the ROV a little bit earlier, because they have a skeleton made of loose framework of spicules, so they'll fall apart when the sponge dies, as opposed to staying kind of embedded together or woven together.
Interesting.
Oh, we have another
Ooh.
black coral, so the
nice.
chrysopathes that we were seeing earlier. And that we collected. Pilot, would you mind if we get a zoom on these little red circles? We've been seeing them all day, and I don't think we've had a real zoom on them to get a closer look. Oh, there went one.
Ah, that's so funny. I love how they've just been doing that all day. In and out. Oh, cool. Oh,
There it goes.
it's shy. Oh,
I'm not
So
quite sure.
close.
They're hard for me to ID. I can't decide what I think it is. But maybe somebody in the chat, or Jenny or Abby, who've worked in this area a lot, if you guys have an ID for the red little circles that seem kind of anemone-like, but also maybe possibly a worm. Oh, there's kind of a bush right on the lower left corner. We can start with a snap zoom, see.
Oh, Annie thinks that those red little guys are likely tube worms,
Okay.
but is also, she said, also can't really ID, so not quite sure.
Oh, this looks like a big colony of hydroids. So a hydrozoan, which is a type of cnidarian, but not corals. You see them kind of all over the place. This is just a pretty dense colony. That's good. Thanks, Pilot. Oh, yeah, I think they are tube worms.
Oh, there's a sea star in there.
Oh,
Kind of hiding.
I think there's also one. See one right under that.
Oh, yeah.
Yeah, we have a sea star kind of hiding under this ledge here. I think it's a sea star.
Yeah, I can't tell.
Oh, no, I don't think so. It has siphons.
Yeah. Oh, curious.
Chris Moss says this is a tunicate. And we have a really nice view of that squat lobster also.
Mm-hmm.
The same one we saw the other day.
Is that shiny portion in the upper right associated with the tunicate?
No, I think that's it could be a brachiopod shell or some type of,
just like embedded.
yeah.
Oh, huh.
Yeah, I think it's, I don't know, but I think it's a shell of some kind.
Yeah, that sheen looks very...
Iridescent.
Yeah.
Yeah, Jenny Waddell says she thinks it's a shell also.
Ooh, there's a recently eroded feature on the bottom left. It's kind of cool. Featuring some larger holes. We're good to keep traversing over.
Oh,
is that
to the right, we've got some corals. Or right in front of us, there's somebody.
It's kind of like waving around in the current.
Maybe just a snap zoom, yeah. Yeah, this is like
neat.
the black coral that we've... I don't know if it's the same one, but it is a black coral. It's not as dense as the others. But that's good. Thank you.
More cobbles with life.
Oh, we have more of those sea cucumbers right there on the left.
Oh, yeah, the branching ones.
Yeah, we have those more of those soulless holotherians, and then some of this paragorgia. There's
another one of those sponges.
Yeah, another
we've
sample
sampled.
of that sponge that we sampled just a bit ago. Is it worth trying another rock, like that black rock?
I think that these are going to be
Okay.
embedded, yeah.
Just trying to get you a rock.
Oh, yeah, no, I appreciate it. Yeah, and I also think these cobbles are some incorporated glacial moraine, so I don't know how telling they would be for the environment.
Can we take a snap zoom on that pink coral on the left? Yes. Yeah, looks to be that. We'll take a close look, but that same genus we've been seeing. Yeah, so that same octocoral has those same shrimp hanging out. That's great. Thanks, Pilot.
Oh, that's fun.
One of those capryllid amphipods hanging off the side. Oh, and another one.
Oh, cool.
Yeah, they use their hind legs to like wrap around. There's some little crabs down there.
Yeah.
Yeah, those capryllid amphipods use their hind legs to wrap around. So when you if you collect a coral with them on, you have to like very delicately pick their legs off.
Oh, really?
Yeah, they're pretty secure.
Oh, dang. Yeah, so it looks like we've approached kind of the top of this sloped region that we've been investigating. So I think the ROV team is going to just continue west, kind of laterally along this rock face. But it'll likely be a little bit less steep than what we've been seeing earlier. So much sediment. Yeah, very angular and strange.
Some more sponge on the side of that wall also.
Oh, yeah.
Here comes a crab.
Ooh, crab.
So here we go. I'm going to go around this edge.
What could be here?
We haven't seen any octopus today.
No, we haven't. It's surprising, yeah, because yesterday we saw like several in one place. I guess there are a lot of nooks and crannies for them to hide if they were here.
That's true, too. There was a sea star just, sorry, is it possible to get a zoom on the sea star we just passed? Yeah. Thanks. Yes, that one.
The way this sea star is oriented, it almost looks like it's like walking forward.
And while we have the ability to stream to shore, there's a slight lag, so just a couple seconds where sometimes it takes a few seconds for scientists on shore to be able to see and then identify something. So when we're looking at it on board, it can feel like, okay, we haven't heard anybody chime in, but it's also that there's a little bit of delay. Chris Moss says this is another one of these amphiurasters that he's never seen this many of them before.
Interesting.
So that's cool. And one thing that was really, I learned today was that since the inception of the Okeanos Explorer in this program, there have been over 350 species described from what's been observed and collected by the Okeanos Explorer.
Oh, wow.
Which is a really great addition to science and being able to really talk about the value of collections. So sometimes we get asked why we collect organisms, and it's because you can't really describe a species or identify if something is a new species or even a new genus or family if you don't have that physical biological specimen in hand to do really up-close analysis or genetic analysis. And so being able to get the high-quality in situ or in environment video in addition to having the physical specimen allows us to really be able to describe species in the best way possible.
Yeah, and once we have these samples on board, we're able to preserve them and then distribute them to, I believe, the biological samples go to the Smithsonian, and the geologic samples go to Oregon State University Rock Repository, so that they can be used by scientists anywhere.
So another kind of conversation that's been happening in the science chat is commenting on how low the dissolved oxygen is for this area. So dissolved oxygen is how much oxygen is in the water for organisms to use or for anything for respiration. And so it's been around 0.3 to 0.4 all day, which is very low. So we're likely in the oxygen minimum zone, which is an area of depth that is where you have really low oxygen, and sometimes that can limit the type of biology that can live there. So that also might be a factor as to why we're not seeing some larger colonies of corals or other types of organisms, is that they just can't tolerate this low oxygen level. Yesterday's dive, we were in the range of like 1.2 milligrams per liter, which is still pretty low, honestly. But we were seeing also a different kind of environment geologically as well as different biological communities yesterday.
Yeah, definitely.
Pilot, when we have a chance, we'll fire all 5 Niskins. Okay. I did. Well, okay.
I haven't really been seeing a lot of great samples, so I think it's okay to keep going. Yes, we are ready.
Yes, we are ready. Number 1, Niskin 1 secured.
Got it.
Legs.
Where?
It's coming up on that. They have that view before we...
Oh, there it is.
He is missing a leg.
Oh, no!
Or they are missing a leg. Yes. Niskin 2 acquired.
Got it. That was number 7. Mm-hmm.
Oh, we have another crab down there that doesn't look so good.
Oh, interesting. It looks, yeah, its legs do not look correct.
Actually, if we can zoom on that lethargic looking, it might be just it might be a molt, so it might be just the leftover.
Oh, cool.
Or maybe he's taking a nap. But I don't think so.
Yeah. It's hard traversing these slopes.
I bet. Yeah, I would say that that is a molt, so you can see
neat.
how it's almost a little translucent. Like, it doesn't look like there's flesh inside, and then the eyeball sockets are also, it's either a molt or it's dead.
Yeah, do they molt their eyes?
No, I
Region?
don't. Let's see if
Curious. Yeah, I don't know.
Yeah, Bob
Bobson-
Carney also said possibly a molt.
Interesting.
But so crabs grow an exoskeleton essentially, so they don't have. They molt their whole external
we are
structure
on-
in order to grow.
Yeah,
to
we're on
continue to
number
grow.
9 now. So that was number 8.
Yeah,
Interesting.
I think next time I need to put my head on the desk, I'm going to say I'm doing a crab molt.
Yeah, but Jenna Hill says it looks like a yoga pose. The crab molt. That's funny.
Thanks, pilot. Wait, third Niska. Wait, when did?
Fourth.
Oh, I missed
Okay.
one. Okay, sorry.
I think we were looking at the molt.
Oh, sorry. Ooh, we have a nice big sea star.
Ooh.
I think we've seen these ones today. We have that shaggy sponge. So we've been talking about firing the Niskins, which is a type of bottle that we send down to the seafloor on the back of D2, the ROV, to collect water from a place that we know exactly where it's coming from. So it's a container that goes down with both the bottom and the bottom and the top open, and then we can decide when we want to close that, and the RV pilots can hit a button and it triggers that to close, and so we know exactly where that water is coming from, the depth, as well as all of the oceanographic data like temperature and oxygen and salinity, and then we can bring that up to the surface and filter that water for eDNA, or environmental DNA, which is DNA that's left in the water column from organisms nearby. We have a nice paragorgia and a sponge.
Yeah, and another one of those glacial cobbles, kind of positioned center screen, likely hosting a group of organisms.
Are you ready for Niskin 5, team?
I'm ready. Woohoo!
Niskins acquired.
All 5 Niskins.
It's a nice sponge on the edge there.
Yeah, I feel like we've been passing by a lot of these vase sponges. I don't know if they're the same.
This is what that's what Abby had kind of talked about earlier, where it's hard to ID those without the sample, but they're lysocene sponges. She said there's a couple of different genre that are in the area.
Uh-huh.
And Annie Lindgren had given us some info about the oxygen minimum zone. So she said that you can say that the OMZ, or the oxygen minimum zone, is due to higher productivity in the overlying waters in this part of the ocean. as all that biomass and biodiversity at the surface sinks, aerobic microbes decompose the material, respiring in the process, so using up oxygen in that process. So I think we just have about 10 to 15 minutes left on the bottom.
Yeah, I believe so.
Oh, there goes the sea star. He'll make his way back up.
Oh, yeah, easy.
And like we said, this is the 8th dive for this expedition, but we will be diving again tomorrow, likely in this same canyon. We will find either a new place for a canyon dive, or potentially we'll be looking for a underwater cultural heritage site, but that'll be determined later today after we look at some of the seafloor mapping data. And we'll have another dive probably in a similar depth range, so a nice long dive on the seafloor for tomorrow as well.
And tomorrow will likely be our last dive of the expedition. So if you've been following us, we kind of started off of Astoria, Oregon, worked a little bit down south there, and then slowly kind of migrated our way up north following the margin. And right now we're just landed right outside of the Strait of Juan de Fuca, and later this week, we will be docking in Seattle, and there's going to be a Seattle port event on Saturday, April 29th. I always forget which exact date, but that's where the public will have a chance to see the Okeanos Explorer, and you can take a look at the 2 ROVs that we use, and meet some of the people that are involved in our exploration. And we'll be there, I believe, from 10 a.m. to about 5 p.m. And, yeah, we hope to see you all there.
And you've been listening to us all day, but my name is Alexis Weinig, and I am a biologist with the U.S. Geological Survey.
And my name is Paige Koenig, and I am a geophysicist with Western Washington University.
And we have been sitting in the back row as your science leads, but our front row is occupied by the members of the Global Foundation for Ocean Exploration, which is a nonprofit that maintains the vehicles, so the ROVs, and then also is the team that conducts our video engineering. So acquiring all those really great close-ups and zooms, and creating the video products after the dives as well. This is just the first expedition for NOAA Ocean Exploration and the Okeanos Explorer this season, so there will be more ROV cruises to come, more ROV expeditions to come as the season continues. There'll be a expedition in the Aleutian Islands off of Alaska, as well as around Dutch Harbor off of Alaska. And lots of mapping happening, lots of seafloor mapping.
Oh yeah, as always.
We've had 2 days full of these jellyfish.
Yeah, they've been really lingering around. They're really beautiful to see, and you can see them really well in the Sirius kind of overhead cam, kind of looking over D2. You can watch them kind of float around. Very relaxing. And today we started at about 1,000 meters depth and right now we've kind of traversed up the Nitinat Canyon slope to about 898 meters. So, long trek.
Yeah, that's great. Thank you.
Yeah, thanks.
So our ROV navigators let us know we have about 5 minutes left in this dive. So, I mean, I would love to look at this coral. If
One
that's
last
a possibility.
look.
Thanks. Yeah, you're doing great.
And I'll also mention, just so I don't forget, that we are just outside of the Olympic Coast National Marine Sanctuary. Well, the Nitinat Canyon is protected by the Olympic Coast National Marine Sanctuary where we're at, and this canyon is also really important to the Makah tribe located onshore, and so this science is not only valuable to the people involved in this expedition, but also the people on shore who call this place home.
Yeah, it looks like another black coral. This Chrysopathes that we were able to sample earlier today. We've seen a lot of these along today's trek, and there looks like maybe a brittle star is also calling this coral home.
Oh, cool. Really bushy again.
Yeah, lots of polyps all along these branches, and a lot of different, a lot of dense branching. So you have the main axis, and then you have the branches coming off, and then lots of little branches on the branches. And then even when you look at the base, you have those longer tentacles. So I can't be for sure why they do that, but this has been something that has been observed with other black corals, where you have much longer tentacles around the polyps that are at the base. So it's interesting to see that here too. And we got a really great collection of one of these earlier today that we'll be able to use for a number of different things. We were able to collect the actual base of the coral, which for black corals can be used to date them with radiometric dating, so we can figure out exactly how old that coral colony is, which is really important information for trying to understand the ecology of a system like this. Thanks, Pilot. That's great.
A perfect last specimen to look at before we ascend.
We ended on a black coral yesterday, and we ended on a black coral today.
Oh, wow.
We also were accompanied by those thorny head fish all day.
Yeah. They just had to say its final goodbye.
Still pretty steep. It's just so muddy here, or like there's just so much sediment.
Oh yeah, it's super eroded here.
I do find it really fascinating what you and Jenna Hill were talking about earlier today with the differences in this canyon and where we were yesterday.
Quinault Canyon.
With the two different like types of, like why we're seeing the geology there versus here, and the earthquake activity. I think that explains to me as a simple biologist a lot more about why we have the topography we have between those two places.
Yeah, and it's really interesting thinking about the different types of species distribution that we saw as well. You know, like this place experiences, or in Nitinat Canyon, experiences a lot of earthquakes, which can displace the sediment and kind of erode away the homes for a lot of these creatures, and that kind of limits our ability to see some of the kind of larger animals that we saw in the non-tectonic activity in Quinault Canyon. So full connection to geology, seismology, biology.
Awesome. Thank you, Nev.
Thank you.
And if you're on the line, we are going to do a pre-dive call on our way up, so you should have that teleconference number, and we will hear from you soon. But other than that, we will do a quick sign-off to say thanks for watching today and for exploring with us. We have been in Nitinat Canyon off of Washington, and we worked our way up from around 1,100 meters to around 900 meters.
Oh, wow. Yeah, thanks for tuning in and joining us, and thank you to our onshore science team for their awesome contributions.